Hello, hello, hello. Welcome everybody. Welcome to stream. Hello. Hello. Welcome. Been a little while since I last talked to you all. Hello, welcome. Hello, Dreamless Sleep. Although I guess reading your comment, you may appear to be dreamless and sleepless for now. Hello, James. Hello, Isotan. And DR Lava Cookies. Hello, Seed. Welcome, welcome. Welcome to this PowerPoint presentation on cell biology, which is one of the most central disciplines within the field of biology I think we have here. Hello, Gold. Welcome, welcome. And if you haven't already, you can help me decide on what merch to pump out by following the link in the description as well as in the pinned comment. So if you are thinking about maybe supporting the channel by buying some merch, you have a chance to sort of shape what form that takes on by, again, following those links. I've got these two ideas here, besides just stickers of the Find the Neutrophil Fiverse, the complements pathway. This is the idea, I think, for some of the more complex designs. But if you're happy with just the stickers and magnets, that's totally cool too. I just kind of want to know what people are interested in with regards to what you would want to buy from me. I think that's pretty cool. It's also one of the charity incentives that we met for the cancer stream that we did an entire 9 months ago, which is actually pretty insane to think about. All right, so let's go ahead and let's just get started, why don't we? Let's start talking about cellular biology. All right, so Cell Biology 101 here. All of these assets are taken from Wikimedia or from open access papers, so I will be relying heavily on diagrams obtained from there. So again, we're talking about the biology of the cell. So what is cell biology? It's the biology of the cell. This is the most obvious explanation here. But it has to be this vague, I think, because cell biology is so vastly varied, right? It's incredibly vast. It covers so many different things that there's no way that I do an actual 101 on the whole field of cell biology. So instead, we're going to format this with basically everything that I personally remember about cell biology and then some cool vignettes, I think. That is the best way that I thought to do this, because I was just like, man, there's a lot that the cell does, and it's really impossible to talk about all of it in one stream. But I think the first thing that I want to do here is to actually put cell biology within the context of the spectrum of biology. And I figured that'd be kind of fun to do. And I think it'd be cool to start off with biophysics, right? So just sort of identifying where in the universe of biology cell biology is kind of important. So we can talk about where the fuzzy boundaries of what this term means actually lies. And the first category that I wanted to talk about briefly was biophysics, right? This is, as you can imagine, maybe the start of understanding life in a molecular context. And this is something that I really want to do a 101 of, but I'm not entirely sure when I would do it. And the reason why that is because I'm just not that good at this. My cell biology class went a lot better than my biophysics class. But if you want an idea of what a biophysicist might think about, it's things like, how do ions interact? Like, what forces are active on a biological molecule, right? So you can think about the electrostatic force between an ion and the head of a phospholipid molecule. That's something that a biophysicist might be interested in. You can think about forces literally being forces, like the number of newtons that a motor protein pulls on a microtubule is also something that a biophysicist might be interested in. There are computational and informational aspects of biophysics, like the intrinsic complexity of a particular biomolecule. All those things are interesting for a biophysics to think about. All right. Chemistry, biochemistry, is a little further down in abstraction, where we're thinking about reactions, right? Binding, catalysis, and I actually did a biochemistry 101 in stream before with clockwork. or at least I think I did Proteins 101 with Clockwork. I don't remember if I did Biochem with him too. But this idea is this is now leading into the field of the chemistry. You're talking about thermodynamics, reaction rates, the binding of proteins and other biomolecules, et cetera, folding structure. And then from there, we get cell biology, right? And cell biology zooms out to the perspective of the cell. And I know that I've said that a couple of times so far, right? It seems obvious, but I think I want to spell it out very clearly here that we are moving outside of the realm of just looking at interactions between individual biomolecules and are now looking at a perspective of a cell as a complete thing. So cell biology is really everything that this thing does. And turns out that can be kind of difficult to describe because this thing does a lot of different things. Listening to cell bio while studying for bioinformatics, my brain is going to be fried. Oh, don't do that. Bioinformatics, very, very cool. A class that I've had a lot of trouble with myself. But I did a collab with Resta on that topic, and I found it to be quite interesting and chewable, digestible, I would say. I forgot what enthalpy means. I think enthalpy is the change in the... Oh my gosh, it's been a while for me too. Entropy is disorder, and I think enthalpy is heat, like chemical heat. Someone can elaborate on that much better in the chat. All right. So I personally, if I were to try and describe a unifying concept about cell biology, it would be thinking of the cell as something that can compute inputs and outputs, right? So the cell receives a signal either from outside of itself or inside of itself, and it reacts to that signal and produces an output. I think if I were to draw one very broad brush around the entirety of cell biology, it would be this concept. And I think it's not a bad idea to view cell biology in this way. So here's a little exercise for you, chat. So if we consider the cell to be a computational unit that converts glucose as an input and outputs ATP, what is this process called? Anyone have a guess or can just flat out recall, what is this process? The process of converting glucose, a sugar molecule, to usable cellular fuel in the form of ATP. While I take a sip of my tea here. Oh, it's good. I realize I need to be hydrating more because after the 4-hour research hell stream with Mylan, my voice was not doing well after that. Mitochondriaing, ATP synthesis, respiration, glycolysis, everything here, that's all pretty legit, right? So we can think about this process, input and output, and we can label that process with all of the things that chat has said, which is very cool. Although you can think of this in terms of biochemistry as well. It doesn't have to be solely in the bucket of cell biology, but this is one of the ways that a cell can take an input, glucose, and turn it into an output, ATP. What about this? What about this? A computation that involves taking a growth factor and converting it into the process of replication. What is this process called? Something with Krebs cycle 2? Yeah, exactly. Cellular respiration. That'll do. Was it cell breathing? I mean, yeah, you could call it that. Signaling would be one way to describe this, but what is the actual name that we give this? Or one of the names that we give this. There are a couple of correct answers that I'm looking for here. Taking growth factor as an input and converting it to replication as an output. What do we call that? So C says mitosis. That is certainly one process by which that happens. Epigenetics, not quite, right? Epigenetics is the process by which DNA is modified to affect expression of mRNA and subsequently protein. So not quite. Mitosis is one of the correct answers. Can anyone think of another way to describe this process? The process of reacting to a growth factor and then replicating. Mitosis is definitely in this bucket as well. Growth factor induced mitosis, very technically accurate. Meiosis would maybe count, although I don't remember how meiosis... I think meiosis depends on each individual organism. I don't know off the top of my head if this fits for every organism in terms of meiosis. Exiting G0, what is G0 a part of? That's what I'm fishing for. So G0 being one of the gap phases of something, a term that I'm fishing for. G0 is a part of that. Cell cycle, thank you. Cell cycle, exactly. Cell proliferation and growth, all very, very good answers, right? So so that's the cell cycle. And this is, I think, the last one I have here. So this could also be described as something to do with cell biology, is taking the input of a viral infection and then dying. Turns out that's also something that is studied in cell biology, right? So what would this process be called? Taking the input of viral infection and then dying as a response, right? This is pushing maybe the definition of input and output, but I still think it's valid to view it in this way. Apoptosis, very, very good. Anything else besides apoptosis? This is a challenge question for y'all. Apoptosis is definitely an option. Necrosis is also an option, right? What is the broad term for these things, I should say? Lytic cycle, also true if the cell that you are talking about is a bacterium. Although, you know, most of what I do is mammal stuff on stream. But yeah, Guy, you're absolutely right. If we're talking about bacteria, we're often in the lytic cycle. Not quite as common for mammalian cells, though. So what umbrella term does apoptosis, necrosis, what umbrella term do we have for that? I'm actually not sure if I put it even in the slide, but I just thought of it now. What process is apoptosis? Cell death? I mean, technically true, but what kind of cell death? What kind of cell death? It is cell death, but controlled death. Very close. Yes. Controlled death, or in other words, programmed cell death, right? This includes apoptosis, necroptosis, pyroptosis. Lysis is not really a programmed form of cell death, but we can think of the program that the computer of the cell runs as being we can think of apoptosis, necroptosis, pyroptosis as being a program that the cell runs when it receives the viral input and it outputs its own death, right? Programmed, it's a computer. Yeah, I like to think of cells as computers. I usually don't have a lot of people who have watched my channel know my feelings on techifying biology and how you should do it very carefully, but I'm actually totally okay with viewing the cell as a mini computer because it can take inputs and then transform them into outputs, and I think that's totally valid way, as long as you don't. I don't know how you could view that incorrectly. I don't know how you could corrupt that concept, so I'm just going to say that that's pretty okay to think about. Hey Astro, welcome. Yeah, so here's my agenda in terms of trying to give you an overview of cell biology. This is not something that you should this is true for any stream that I do. There are YouTubers out there who specifically make content for you to study from, and this is not it. This is more me just saying things that I like about cell biology, hopefully getting you interested as well. So if you are a biology student, this is not going to be helpful for you outside of being maybe interesting. It's interesting maybe to hear some of these vignettes, but I really desire to, that's a weird way of saying it, I really want to cater these one-on-ones to a general audience, to get you interested and informed as to what a general field is like, as opposed to being study material. And I say that as a hedge because this is a very messy guide to cell biology. It's mostly just a tour around the places that I like. So don't take too much out of this, I think. All right. Maybe mutation isn't changing the codes, as mutation doesn't always destroy stuff, but typo and code will break everything. That, I would say, you could think of in a cell biological context, but I think geneticists would be more interested in that, or biochemists, because cell biology, I'm not saying that's not included in cell biology, because, like, let's be real, almost everything is cell biology if you're talking about molecular biology. But where I would fit more comfortably, where that bucket would be, like, I would think that more as in like biochemical terms and then feeds into cell biology eventually. But to me, thinking about mutation is a bit removed from cell bio. Not totally removed, of course, because all these categories are fuzzy, but for me, I think of that more as genetics. So we'll do a super fast organelle review, because to understand cell biology, you just kind of have to know the stuff that's inside of a cell. And then we'll do a super brief overview of all the processes I remembered that I learned. I won't be talking about them in any detail, but if I have interesting vignettes, I'll share them. So we'll speed run through that. And then we'll get into some more crunchy stuff, like the flow of information, which is a consistent theme throughout cell biology. And then we'll talk about one of the most involved things that you'll learn in class, which is the cell cycle. And then we'll finally end off with a modern vignette, the blebisome, which is a brand new thing discovered in February of this year, showing how the field of cell biology is nowhere near complete, that there's always new and interesting stuff that is being discovered. All right, let's get into it. Okay, so organelle review, the first big one that I think we should review is the membrane, right? This is such a fundamental piece of the cell that it kind of defines it, right? I mean, you don't have a cell without clear boundaries, and so this is an excellent place to start. The whatisome, the blebisome, great name, I know. But it actually, like, the word bleb actually has a very precise contextual biological meaning, which we'll get into when we get to that vignette. But for cells that harbor life, they are made of phospholipid bilayers that provides a barrier between a cell's insides and its outsides, right? And so, you know, this is not a universal plan for all living cells. Some cells have more than one phospholipid bilayer. Some cells have an additional cell wall on top of that made of various components. But this is the general unit of the cell, right? You gotta define where the cell ends and where it begins. And so this membrane is really, really important for keeping the cell's insides in and its outsides out, right? And this membrane is also studded with a lot of different important proteins that help the cell react to its environment, as well as maintain its own integrity and do all sorts of other things, right? But I think what is interesting to think about, an interesting takeaway, is that a membrane had to have happened, right, for the origin of life, right? Because before we had actual cells, we were all just in the primordial soup, a bunch of chemicals and proteins and RNA, or, you know, debatably, depending on who you believe in terms of the origin of life. But before any of that gets to be called life, the things that live inside the cell had to be concentrated in a high enough concentration for productive chemical reactions to actually occur, right? It's not just good enough that the biomolecules of life exist out in that primordial soup. They all have to be close enough to one another for there to be a non-zero chance that they interact. And I think thinking of the membrane as a container for all of the stuff to really make sure that things happen in a productive way is an interesting way to view the cell, I think. Membrane gang, true, best organelle. I think so, in a kind of way, in a way where this is where it all begins, right? Before you get fancier things like mitochondria and the nucleus, you have to first start off with a container, a bucket. All right, so next organelle, we have the nucleus. So this is, of course, the command center, here labeled in blue with the ever-present DAPI stain. This is the command center. It houses the DNA, which codes for the blueprints of life, right? If you want to hear more about that, you can watch my stream on genetics, but I won't really dwell on it here. And then we have just outside the nucleus, in labeled 5 here, we have the endoplasmic reticulum. Let me move myself out of the way here. The endoplasmic reticulum. So this organelle is, I think, underappreciated in the wider context of cultural understanding of biology, right? These are organelles that are continuous with the nucleus. So we think of like the nucleus and the endoplasmic reticulum as different things, but they don't really have a strict border around each other, right? They are connected. And the endoplasmic reticulum is made of rough components labeled here in 5, and then smooth components labeled here in 8. And these places have different functions. There are a lot of things that this organelle does. The rough ER is studded with ribosomes, which are the cell's protein factories. These are protein production units that read messenger RNA and spit out protein. And then the smooth ER doesn't contain ribosomes. This is an area that is important for lipid synthesis, especially important for cells that detoxify things, specifically in the liver. This organelle does like a billion different things, impossible to get through all of it, but it also acts as a sorting relay for proteins, namely secreted and membrane integral proteins. And it's also a sensor of cell stress, which if you watched my Prion video, you would already know, right? The unfolded the what is it? Unfolded protein response, the UPR. This is a cellular program, again, one of the many programs that the cell can run that detects whether or not folding is happening properly. And if a cell is not healthy enough to be folding protein properly, it will activate this attempt to fix this problem, right? Yeah, this organelle does like a billion different things. I also remember that they're calcium regulators inside the cell. Right, I think I remember that too, right? Or am I getting that confused with Golgi? I think it's the ER where they are calcium regulators as well. Just so many different things that this thing does. Are there also ribosomes in the cytoplasm or just the rough ER? There are also ribosomes in the cytoplasm. They do not have to be in the ER, and we'll actually talk about that later on. Are those remnants of when eukaryotic cells first appeared? I'm not entirely sure what you mean by that, Seed. Or are you talking about the coacervates, which I don't actually know too much about? Then we have the ribosome, right? So it makes logical sense to talk about protein synthesis after detailing what the endoplasmic reticulum does. This is the catalytic RNA protein complex that consists of two subunits, and these two pieces come together and actually form the thing that can convert mRNA to protein. And so in green and yellow here, we have the large subunit and the small subunit. The large subunit binds transfer RNA, which is another type of RNA that can bind individual amino acids. This is what's actually doing the knitting of the protein, right? You see on top there are these like little things that are coming in and dumping in amino acids that get pushed out the bottom. And the bottom subunit, the small one, reads messenger RNA. So there are two pieces of this machine, and they both, one functions to read the mRNA and the other functions to bring in the appropriate amino acid as it's being stitched. Here in this animation, you can actually see the ribosome being docked into the membranous component of the rough endoplasmic reticulum. The endoplasmic reticulum. I hear that the cell nucleus formed by invaginations in the membrane folding around DNA. I actually don't know. I don't remember enough about how that works. Full disclosure, I thought cell biology was very, very hard, considering there were so many things to remember. So that might be true. I just don't remember it. All right. So then we have the Golgi. If you are a long-term viewer of Phytoneutrophil, this might not be, you might remember that I have a distaste for the Golgi. Before that, I'll read Ali's comment. I find it interesting that ribosomes are made of rRNA, using tRNA to transcribe mRNA. It is interesting, but it's not wholly unsurprising, right? Because how do you read how are you able to like read and bind mRNA? One potential way to do it is by also being made of RNA yourself. So I think it's neat the way that the ribosome is constructed. All right, so the Golgi, right? So this is the organelle that is like the postman. It is like the vesicular transport organelle, where it can it is a central node by which things move through to get to their final destination. The reason why I'm a Golgi hater is because I found it particularly difficult to learn while I was taking cell biology myself. It's pretty complicated the way that this thing works, which we're not going to get into today. But this is an organelle that is not deserving of my ire because it's involved in all sorts of interesting immune processes as well, including the generation of lysosomal vesicles, which are these acid pockets that degrade proteins that are important for protein recycling as well as processing vesicles that are meant to be secreted out from the cell. So cool organelle, I just didn't like learning about it. All right, mitochondria, you already know what you want to say. It's the powerhouse of the cell. Producer of ATP and oxidative stress, a big reason why people age at all and develop certain conditions are because of the mitochondria. We signed a deal with this ancient bacteria a gajillion years ago where we would house them and they would provide us with tasty energy, right? But this is such an interesting organelle, not only because of its origins, but also because of the myriad of things that this also does, right? So not only does it provide us with energy, it's also one of the first line immune surveillance organelles that we have in this cell, which is something that I learned not that long ago, surprisingly. And it's also got its own DNA, which is also very cool. You inherit your mitochondrial DNA from your mother because sperm don't really contribute their mitochondria to the egg, the zygote making process. So you can trace your matrilineal our mitochondrial lineage through your matrilineal line, which I think is also very cool. Tiny cells in our cells, exactly. All right, and we have the cytoskeleton. This is the bones of the cell, which is, I think, very, very cool and interesting because I like pretty pictures. And for those other organelles who don't really get pretty pictures quite as often, here we see the 3 different types of proteins that can contribute to a cell structure. We have the tubulin, the microtubules of the cell, which are outlined here in green. I know if you're colorblind, I'm sorry. Biologists have a terrible habit of doing red-green coloring, but these, I'll try to mark that out on the slide here, but these strings here, these are the tubulins, these are microtubules, these are rigid and big structures. You have the actin subunits in red here, which are very important for membrane dynamics, and then you have intermediate filaments, which kind of do a lot of things that are varied and impossible to summarize. I love the cytoskeleton. I did my undergraduate research in cytoskeleton stuff, so I have a fond place for the cytoskeleton in my heart, and it is a surprisingly dynamic system, which we're going to see later on in this presentation. Right, your cells are very much alive, even though these are bones. These are like bones made of jelly sometimes, which I think is really, really cool. The gray, the grayer, and the grayest. Okay, if you're a budding biologist out there, use blue and orange and magenta, I think. There are colorblind friendly color schemes out there that people have just been very stubborn to adapt. Yeah, and the cytoskeleton, it underpins a lot of what the cell does too, which I think is really, really fun. Okay, here's a video that I think is cool. Just showing you how dynamic the cytoskeleton actually is, which I don't think you get in a typical high school education. So here you can actually see, labeled here is, I think, GFP labeled F actin, right? So this is an epithelial cell. I think this is a kidney cell just on a dish. And you can see just existing that the actin, you can even get like, we'll watch it again. You can even get like a very clear indication of where the membranes are, even though the membrane is not labeled, right? But this looks like a cell because actin is such an important part of ensuring membrane dynamics, which I think is really, really cool. And also, again, I love video and I love pictures, and it's always really, really fun to see. All right, and then we have the centrioles, which are just kind of tacked on here because I always forget these things exist. These are tubulin organizing centers, so they are kind of adjacent conceptually to the cytoskeleton. They organize the spindle and the aster for cell division, so if you took a high school class recently, you'll know all about the centriole, even though I forgot most of what I knew about it. They look kind of cursed to me. Honestly, they look very cursed to me all the time. And they kind of remind me of Forbidden Twizzlers, and I don't have that much to say about these guys. Okay. So that's a very, very quick introduction to just the ingredients of the cell, right? And now I want to talk about things that I think are a little more fun to talk about, which is the processes, right? I talked a lot about how you can think of the cell as a computer that takes an input and then spits out an output. So I just want to give a little tour of the things that I remember about cell bio, about learning in cell bio, right? Obviously, if I were to do everything, that would be a very, very long stream and possibly impossible, but just the things that I remember learning in classes. Now I'm going to be so prepared for cell bio next semester. I hope you will be. I think that's, like, even though I say don't use this to study, if you've never taken a biology class before, this is a nice overview, a nice little appetizer for you to look forward to learning about when you actually learn it formally. So here's some topics, right? So how is DNA copied and read, which overlaps quite heavily with biochemistry and genetics. By the way, Clockwork Bio did an amazing video on the replicon, which you should definitely watch. And that stuff is very nitty gritty with the dance that the proteins do, which is a little out of my scope for the content that I like to cover, but it is really quite excellent and very beautiful the way that the system works. There's also, you know, on the end of like how is DNA read and made into RNA, there's also the other end of that spectrum, which is how is protein made, which is an entire system by which, you know, mRNA leaves a nucleus, docks into the ribosome. The ribosome also has a bunch of processes within it that govern how these tRNAs are depositing their amino acids and so on and so forth. Like, this is an entire week just learning how this happens, which I think is really, really tough. And then this is kind of interesting. This is something I don't think that you get in an intro bio class all too often. How do proteins know where to go? And if you've taken biology before, don't answer, right? I want to give the non-biologist, or if you've taken biology like a billion years ago, this is a fun way to get back into thinking about all sorts of the clever things that the cell actually does. So how does a protein know where to go, right? We know that proteins have to have some kind of information, or there has to be some information encoded somewhere in this cell that enables a protein to understand where it should be. So how does that happen? Again, non-biologist answer first. I'm doing an undergrad degree in bio, but I pushed all the cool biology stuff to the back and started with chemistry and the boring stuff. That's not a bad way to do it, honestly, because having solid foundations in the like more physical side of biology is an excellent mirror, or an excellent window to understand some of this more complex stuff. Cell bio taught me how to spell phosphatidyl ethanolamine. Oh gosh. Yeah, a lot of long words here. Born for biology, forced to do chemistry. There's overlap. You can always choose to do a biology topic while using the skills you learned in chemistry, which is, you know, a vast and interesting field as well. I just did a chemistry lecture to school kids today. That's awesome to see. I love that. Chemistry is one of the more, I think, exciting things for kids to learn. I think biology is a bit of a harder sell. All right, so yeah, so how do you know? How does a protein know where to go? Any non-biologists want to take a shot at this question? I remember a mutation where the protein doesn't remember where to go, something about a lipid side. That's, yeah, okay. That's hovering around the correct answer. But it's far enough that I'm still going to wait for other people to chime in. Any guesses? And this is where I think a lot of learning actually happens. If you've never taken biology before, forcing yourself to come up with something, even if it's wrong, is still a very mentally useful activity. Right? It is still something that will train your brain to tackle new and interesting problems. So how does a protein know where to go? Molecular polarity. That is an interesting idea. So the idea that molecules are different on one end than the other, I'd say that plays a role, I guess you could say. Too many biologists watching? Maybe. Hold on, let me take a poll. How many of you are actually biologists and watching a 101 for cell biology? Are you a biologist? That's so funny that if you are a biologist and you're watching this stream, I think that's cool, and I do enjoy that. Not to say that you can't, right? I just think it's I think it's fun that you would enjoy this kind of 101 content in some way. It's a good refresher. All right. So it looks like we're like 50-50. 50-50. Oh, yeah. Okay, the split is pretty even between biologists and non-biologists. I'm just listening in the background while dying 500 times to a Silksong boss. More power to you. More power to you. I know part of the answer, but is it the same mechanism in all cells? For small prokaryotes, I'd expect Brownian motion to do the work. Ooh, that's an excellent point, Guy. I am specifically referring to the biology of the mammalian cell, because that's where I have most of my expertise. Bacteria, I'm not quite as sure of. I wouldn't call myself a biologist. I have a bachelor's degree in bioengineering, and I went to work in farm due to money for loan/rent. Very, very cool. No, that's awesome. I mean, you know, if you're a biological engineer, bioengineer. Oh, I see. Right, because there are different definitions of biological engineering and bioengineering. I see. Fair enough. I haven't taken biology since secondary school. I vaguely remember them having some kind of bit sticking out that another thing grabs. Oh, very cool, math. surprisingly good memory, I would say, because that is absolutely part of the story. Are they like molecular cars? That's also part of the story, but not in the way that I phrased this question. All right. Very, very cool. Very, very good memories to all of you. So let's end that poll and continue on. No reason you can't, as a full-blown biologist, engage in baby biology to recover some self-esteem or for fun. That is fair too. That is fair. So again, this is, Guy made an excellent point. This is for eukaryotic cells. Okay. Bacteria probably have their own protein localization thing, but this is for eukaryotic cells. The information of where a protein should go on a vague sense, right? This does not cover the entirety of localization. But in a broad stroke, the answer is signal peptides. This is information that is hard-coded in a protein's genetic code. So the DNA that codes for this protein will make a piece of mRNA that then gets converted to a piece of the protein called a signal peptide. And there are a couple of different peptides that can be encoded that will tell a protein where to go for the first step, right? So once a ribosomal protein, once the process for protein synthesis starts, if the protein is destined for the membrane or outside of the membrane, it'll come with it a piece of a peptide that instructs the growing strand to help the ribosome anchor into the rough endoplasmic reticulum, where they can then be shunted off into the rest of the sorting system, right? So a couple of you had vague recollections that this is how it works, and I commend you for remembering that, because that's kind of a challenging thing to remember. So proteins without those signal peptides are often cytosolic, right? So this is one major way where proteins are where you can decipher the information needed to determine where a protein's final destiny is. Of course, this is not complete, but it is a major axis by which this happens. All right. I'm an environmental engineer, and I did have microbiome 101, but nothing too advanced like this. So that's interesting, because I think microbiome doesn't actually... I think because microbiome is not... By definition, it is... I mean, you have like yeast and stuff, I guess, in microbiome. But it's a lot of overlap, but not... But microbiologists will be interested in certain processes over other processes, right? I think I've never taken a microbiome class, but from what I remember, it's more about not about learning the fundamental unit of a prokaryote or a eukaryote, but rather learning about individual stuff, individual species of micro stuff. You can correct me if I'm wrong, again, because I never personally took a dedicated microbiome class. My degree focuses more on being able to maintain temperature, O2, and nutrients for large-scale batches of microbes and the details of how cells work. That's also really cool. I was a purebred biologist up until my last job, where I had to teach biological engineering, and bioreactors, I think, a very, very cool idea that I had not been very privy to before. So that's very, very cool that we have biological engineering representation in chat today. Electronics engineer. I took chemistry, but I really should have taken biology for my science gen ed. I have a lot of regrets on my university education too. I wish I took philosophy, actually. You shouldn't tell Charis Euphranor that, my VTubing philosophy friend, because he'll get a big head about it. But I really wish I took more philosophy. I think that would have been really, really engaging. For my microbiome class, we had to learn 100 different microbes by heart. Yeah, I think that's why I chose not to ever take microbiome. I was more interested in learning how the general thing worked versus learning about how all of them work. Memorizing a bunch of species tracks my expertise. I have a very bad memory for a biologist, which is why I struggled so much in cell bio. I think I would have died in microbiome. All right. Love your vids. Have no idea what you're talking about like 90% of the time, but you sound very cool. Thank you so much, Water Tower. I would say that you probably understand more than you think, because there are levels of understanding, right? I would say for my videos, for the average person that does not have a biology background, I think it's okay that you just understand the vibe of what's going on, which I think my videos do a pretty good job of doing, right? You might not be very familiar with the molecules, et cetera, but if you're okay with just having the vibe down, I think that's perfectly fine, right? I have like friends who have given me feedback. They're like, oh, you know, I don't understand this or that. But I'm like, do you understand the vibe of it? And they generally go yes, because I think this is kind of a tangent in terms of like meta of making content, or making my content, I should say, where I understand that I do like very detailed, relative for YouTube, detailed content, deep dives, as I say repeatedly in my videos. But I think for a certain type of person that is okay with not understanding all the details of everything to be having a good time, I think that's the niche that I'm trying to strike, right? Because if I wander into a physics video and I don't understand everything that's happening, I might be a little upset and not choose to watch it. But those videos aren't geared towards me, right? I think if you like biology enough to be okay with not understanding every little detail, that's actually a perfect place for you. And I'm very happy that you've chosen to watch. Do we know all of the signal peptides possible? Like, are there a billion or a countable number? From what I understand, there are a countable number of categories, right? There's the nuclear localization signal, which tells a protein to head back into the nucleus. There's the signal peptide that directs it to the membrane. I'm sure there are other things that are out there as well. Although it's kind of a vague question because you never know what you're going to discover in the future, right? I've taken an ethics course and it was worth it. It changed me. Yeah, like I think part of what's so beautiful about doing science is that science is kind of a philosophy, and I think philosophy, ethics, morals, thinking about how to think is part of what makes science so interesting. It's not just learning a bunch of facts, it's about learning a way of thought, and I think that is very valuable. My favorite videos are the rabies and the molecular mimicry ones. I think you mean maybe immune evasion, because immune evasion takes advantage of molecular mimicry all the time. Yeah, the rabies video is great. I think that aged well, besides the thing that people jump on me for, which is I said that it's 100% lethal. I said it's basically 100% lethal, but whatever. I did take bioelectronics though, so I know a lot more about nerve cells and how to model them as transmission lines. I know how to not kill someone with electricity, plus best practices for med dev. Oh, that's interesting. I've wanted to do a video about Pikachu for the longest time, but I don't have the expertise, right? I actually tried several times to make a video about how legitimate Pikachu's biology is, to no avail because I'm terrible at E&M. All right. Okay, let's continue, shall we? So we did one part of that, which is how does the protein know where to go, but how does a protein actually get to where they need to go? So I have a picture of the endoplasmic reticulum and the Golgi, and you can see by these arrows that cargo only travels in a certain direction, right? From the ER, you get transport to the Golgi, and then from the Golgi, you get secretion. And then there's also a separate pathway that goes in reverse, right? That is from the membrane and heads back into the cell. But how does the cargo know where to go, right? So the proteins are, they have a certain hook to them that enables them to be placed in the correct bin for sorting, but how do they actually get to their final location? Again, in a broad sense, in a mammalian eukaryotic cell. And again, non-biologists, I'll take your answers. If you're a biologist, you might already know the answer. And I'll go ahead and take the opportunity to catch up on some chat. While the non-biologists hypothesize how the system might work, the fact that there is controlled directional mobility. And as Guy said, there is Brownian motion in the cell, right? Because if there wasn't some strategy for the cell, then you would just have a bunch of biomolecules jiggling around in liquid space and nothing would ever get to where they would need to go in time, unless there's, asterisk, there are some clever things that you can do with Brownian motion that will allow proteins to get to where they need to be. But for the most part, that doesn't work with everything in the cell, right? And not all the time as well. So how does this happen? How do we get such precise trafficking in and out of the cell that's not just reliant on Brownian motion? I think it's very, very interesting. I'm a commercial industrial electrician. I just think it's awesome / amazing stuff. I'm glad to have so many passionate people in the chat who enjoy what they do, because it's not a guarantee. That's just how life shakes out sometimes. There's a protein named Pikachu. There's a protein called Pikachurin, I believe. And then a protein called Sonic Hedgehog, which is a very important developmental gene, which we might see a cameo later on in this stream, if not definitely in the developmental stream. Are there pre-made paths, says Water Tower? Kind of. You probably have to mesh appropriately with a transporter of some kind, which grants admission, and the size is probably a thing. Yeah, that is true. That more has to do with the size of these cargo vesicles. Sure. I've got nothing. It's tricky, isn't it? And it's something that, you know, highlights how clever the cell is. I think even if you try to come up with an example, you can't come up with one. It makes the reveal all that much better. The vesicles are tagged as well. Sometimes, I think. I'm not entirely sure if that is true or not, because there's just so much to remember about cell biology, but it's not the answer that I'm fishing for. They walk to their destination. Interesting thought. They're running on big tubes with a miniature scooter. Also a very, very interesting thought. I remember something about ions being used to help change pressure in the cell or something. That might be on a more macro level, but I don't think that applies specifically to the answer that I'm looking about here. All right, so we'll go ahead and we'll reveal the answer. So again, this is why the cell's cytoskeleton is so goated, right? The cell's cytoskeleton can move directions in a very specific way, right? Kinesin moves along the towards the plus end of a microtubule. You don't have to worry too much about plus versus minus end. Just know that on a microtubule, you know, that bone of the cell, you could say, there is one end that we call plus and then one end that we call minus. And there are motors that are plus end directed and minus end directed. So as long as you have some principle that organizes these microtubules, then you can have a way to have information that dictates what is outside and what is inside. So kinesin will only walk to the plus end for the most part. Dynein will always walk to the inside for the most part, right? And in that way, we can understand that cargo can move in a direction, that it's not just random where it ends up. It does get moved there by, I think Guy called them little scooters, right? I like to call them little guys. Love these proteins. I actually, again, in my undergraduate research, I did a lot of research in kinesin, right? This protein moving along the plus end motor, which I dearly, dearly love. I've seen a video on that walking molecule before. As with most biology students, if you haven't watched Life of the Cell as a biology student, it's like getting baptized, right? Every biology student has to watch Life of the Cell, which is a series done by, I think, Harvard Medical School, which illustrates in beautiful detail all of these things that we are learning about here. And it's aged beautifully, right? That iconic video of kinesin walking along the microtubule has inspired me to become a biologist, right? Because it is just so wonderful, right? Isn't mannose-6 phosphate used to tag the vesicles from the Golgi to be destroyed in the lysosome, or was it the other way around? Again, I don't like the Golgi, so I don't remember. Sounds legit. It sounds all right to me, but yeah, a little too far out in my memory to remember the details of. The do walk, yo, exactly. I mean, I don't remember if you were the one who has yet to take a biology class or has yet to take an upper level biology class, but there is so much joy, I think, in learning biology because of these neat little things that you learn that the cell actually does. It's just so wonderful how these things work out. Like little streets to guide traffic. Yeah, absolutely. You can absolutely think of them as little streets to guide traffic. So how many billions little cute walking molecules do I have in my body? I would say probably an order of trillions. It's trillions or maybe even an order of magnitude higher than that. Although my concept for numbers that big gets kind of fuzzy, so don't take that too seriously. Yeah. Is that the thing you talk about in your rabies video, how it travels through neurons? Absolutely, Water Tower. This is exactly right. Ooh, I'm so glad that you watched the rabies video and can connect it to what I'm teaching today. Absolutely. So dynein is a minus end motor that brings cargo from the outside towards the cell body, right? So if we're looking at this slide, right, you can imagine like rabies entering from here, and then rabies will hitch a ride on dynein, which will walk along microtubules to bring it back to the nucleus, right? So that's retrograde transport, moving back up the neuron, which is very cool. See, like even if you don't understand what's happening in the videos, I'm not saying that's what you said, Water, but like, you know, just speaking generally, I think it's okay. I'm happy if you don't understand everything, as long as you are happy with understanding the vibes of what's happening. How do they know which tubule to take? That is a great question, and it has to do with specificity of the binding of the heads of these proteins, I believe. I think, again, it's been a while since I did Golgi stuff, because this is a lot of Golgi stuff, but I think that there's something that is on the outside of these vesicles that is specific for kinesin or dynein. And there are a couple of kinesins and a couple of dyneins, each of which do different things. So you get a little more resolution on what does what in that way. Plus and minus end are electric charges. I think, I honestly can't say because I don't remember. I don't remember. I do know that tubulin itself is sided. It's got, just like how we have like 5' end and 3' end of DNA, and that indicates positioning on the sugar, we call plus and minus end on a tubulin, but I don't fully remember why, because I whipped this presentation up very, very quickly. So there's that many kinds of kinesin type proteins for each kind of destination. Not quite. There are a couple of different kinesins and a couple of different dyneins, and again, they each do different things and are active at different times depending on what cellular programs have to be active. I'll say that I don't remember enough about the specifics of transport to get into it. That's a 102 kind of thing. Excellent question, though. But it does, again, what I want to leave you with is that this takes care of some dimension of protein localization. It is not complete, though. It is an excellent question. It is a part of the story, but not completely the story. Only like 2 or 3 kinds? I think there are more than that. There are at least 9 kinesins, I think. Oh, it's been so long. I think there are 9 of them. Think. Someone can correct me if I'm wrong, but I think there are quite a few. But yeah, there are other things that determine where a protein ends up that we can talk about during the development stream. Yeah. There's like 30 kinesin genes in humans, I think. Yeah, there are quite a few. There are not like hundreds, but there are quite a few. I think I remember there are at least 9, because I remember calling one of them kinesin 9, or, you know, kinesin family 9 or whatever. Yeah, 30 seems reasonable. Okay. So again, this is another shout out for cytoskeleton stuff, because again, I love cool videos, and this is another cool video. This is a genetic biomarker for the protein CDC42, which is an actin regulator. Again, so if you want to transfer from the question of how does cargo get to where they need to be, well, how do cells get to where they need to be, specifically eukaryotic ones, because bacteria is different. And we come to see that in these videos, we come to see that the cytoskeleton is truly, truly very dynamic, right? And so you can see on the left here, the activity of CDC42, which again is not directly actin, but is an actin regulator. And you can see in this heat map that you have this distinct pattern that then affects this ruffling of this membrane over here, which I think is really, really interesting. And again, if you watched my neutrophil video, you know all about chemotaxis, motility, right? Neutrophils and macrophages who do a lot of moving around will rely very heavily on their very dynamic actin cytoskeleton. And in fact, this next video that I'm going to do, which is a secret that has been teased in the Discord, !Discord if you want access to that, has a lot to do with the pathogen has a lot to do with shutting down the mobility of the cytoskeleton. So that's a little bit of a teaser for you all here. Oh yeah, it builds out these filaments until catastrophe happens. So this is actin. So actin is not treadmilling, but it is directional, and then at a certain concentration it treadmills. Catastrophe happens with tubulin, so the microtubules, so a different cytoskeletal element. All right. Next question, how does a cell divide? Another very cool video. This is actually division happening incorrectly, which I didn't really realize until I cited this source, right? So you have here, and I'm sorry, colorblind people, but you have here in green, you have the microtubules that will attempt to align the chromosomes in time for mitosis, right? So this is another question that cell biology is asking and is figuring out, and has figured out a lot of already. Although in this particular video, it's actually I think a mutant or a treated cell that is struggling to do this correctly. So the chromosomes here are actually struggling to align very nicely along that mitotic plate. Yeah, so if you're a biologist and you don't quite recognize what's happening because it's just a little different, that's probably why. It's kind of struggling to align properly here. Snares and RABG proteins are other important factors that direct the fate of vesicles for anyone who wants to read a bit more. Thank you so much, Lonkil. Yes, that's part of the story that I'm missing, is that snares are part of the story as well. Yeah. Hello, General Mustache. Welcome. So those are just a couple of different overviews, like from 1,000 billion miles up in the air. Every single one of those slides is like a whole week of your life in cell bio, hours of research and cramming for exams, right? And I just summarized them just to give you a little teaser and a refresher, just because I think they're fun, right? Without the stress of actually having to study for an exam, this cell is pretty cool when you don't have to worry about that kind of stuff. But again, I want to return to this idea because it's so fundamental to how I think of the cell. And this is me in particular. I don't know that this is a good idea for other people, but for me, the way that I learned to conceptualize a cell is a little computer that has inputs and outputs, right? So this is a scary looking cell biology pathway figure, and you can see that there are a couple of different inputs that we have here. Wnt and hedgehog. Hey, there's Sonic Hedgehog right here. These are development signals. You have signals that come from the cell's environment, like the extracellular matrix, right? So you can get signals from the perturbations around you, growth factors, chemokines, survival factors, cytokines, right? Inflammation stuff. You have all these different inputs that can be integrated into this complicated circuit, which if you're a computer nerd or an engineer, this might get you kind of excited. Because in the cell is this complex series of circuits that can combinatorically produce certain outputs, whether it be exiting the cell cycle, whether it be entering the cell cycle, whether it be to move along an extracellular matrix, whether it be to, release deadly chemicals if you're a neutrophil, right? All of these different things are different inputs that can then be computed by the cell to form an output, right? It's a gosh darn mess, right? Like, as I said earlier, cell biology was a topic that I struggled quite a bit myself, just because my working memory and my long-term memory are pretty bad. As you can tell, I've forgotten a lot about my basics. I'm sure I could teach myself if I really needed to, but there are better uses for my time than to relearn everything. Instead, I just like to talk about the stuff that I remember, which is often the stuff that left the biggest impression because I thought it was cool. I feel bad for all of you students who still exist out there, right, having to study for your actual exams. But you will know, like, if you're studying for an exam, you will know more about that biology than I would. I'm confident that many people in chat have a better understanding of cell biology than I do on any given day of the week. All right. I think there are also crosstalk on certain channels, like with certain integrins and beta-catenin, which means mechanical strain on the cell can influence its cell fate. Yes, I'm definitely going to talk about that in development, because that is one of the coolest things that I've ever learned in cell and developmental biology. The fact that mechanical strain itself is a signal that if you tug on the membrane in just the right way on a certain receptor, you can influence what that cell becomes, which is kind of insane. I love that. Bioinfo and organ engineering finals in 8 days. Good luck to you, Thomas. Good luck. All right, so today we're just going to talk about from a conceptual perspective, because I think it's cool to do it that way and doesn't require me to reteach myself all of these complicated specifics, just the overall principles of inputs and outputs in a cell. You forget so much if you don't need to learn it. Yeah, exactly. Like, I don't need all of my cell biology at all times. I wish I remembered more of it so I could do a better job teaching it. But, the time it takes to reteach myself cell biology is time that I could be editing the next video. So there are only so many hours in the day. And I am working very hard on this upcoming video, which I'm very, very excited to release. So you can change a cell just by touching it in the wrong way. It's kind of an oversimplification of how it works, right? There are certain receptors that if you tug on them, can cause a change in conformation that will activate a certain program. I'll put it that way. Here we go. All right, so Before I get into that, does anyone recognize this bad boy? So here we have this like very complete cell signaling diagram. Not very complete, but a very broad overview of cell signaling pathways. And here, this is a bit of a meme that has circulated in the biology sphere. So does anyone recognize what pathway this is? So cells have buttons. Again, this is an oversimplification, but you could consider them to have buttons that when they are, maybe it's more correct to say keys on the outside. And when the correct key is put into the correct lock, things happen. I saw Jack Staten had a stroke. Mitochondria stuff. Maybe look a little closer, guy. Maybe a little closer. That image is friends with a certain image of a rat. Ah, Marlon got it. We have little donuts in our cells. I wish. This looks AI generated. Is this AI? Yes. Congratulations for all of those of you who have identified this as AI. You did a better job at reviewing this paper than the Frontiers reviewers did. So here's a warning, not safe for work warning if you are in public. What I'm about to show is pretty graphic. It's the other AI generated image from this very paper. So 3, 2, 1. Yeah, I censored it anyway, just in case, right? But this crazy paper, Cellular Functions of Spermatogonial Stem Cells in Relation to JAK-STAT Pathway, used AI-generated assets, including this rat with what looks to be quite a huge reproductive organ. Just insane. And if we take a closer look at what this pathway is, like this is nonsense. It's fun nonsense. It went to a paper, it went into a journal with a very bad reputation. It's Frontiers. I would say that Frontiers is not without merit, right? Actually, a lot of open access review papers are published in Frontiers, but they are not known for their quality control. Yeah, so let's just have a look here. So if we go back to our very legitimate pathway here, we see that some cytokines activate the JAK-STAT pathway, right? JAK is here, STAT is here. And we'll notice in this AI image, things are just a little bit different, right? You have STAT and JAK activating JAK and JAK activating JAK. We've also got signal binding to the resuscitate. Freaking classic, dude. We have Tramoniacion of Junicoprins. Ooh, but here we have stats clarification. I love this figure so much. This figure's insane. I love this. It's so good. I love how frequent JAK and STAT both appear here, even though it's not this way at all. And I thought Frontiers is a good journal. It's nice if you want reviews that you will have to take with a grain of salt every time you read one, right? Trump notifications, yeah. No, I love this figure. It's great. JAK-STAT, JAK-STAT. Oh, it's so good. I love this. I could stare at this all day. All right, but we must continue. We must. Okay, so how is a cell like a computer? Well, a computer takes an input and puts out an output, right? It's a very useful thing for a computer to do. How does a cell do it, however? A cell, and again, this can be an external or an internal signal, but I think most frequently in cell biology classes, we'll learn about external signals. So the general case for this is that you have a receptor on the outside of a cell, and that binds to a ligand, either a growth factor signal, maybe a signal that an immune cell released, maybe, a cell division signal, whatever, some sort of signal that binds to some sort of receptor in a cell. That receptor will change shape in a very specific way, which will activate its catalytic potential, right? So some biochemistry will happen, and this will catalyze some enzymatic reactions that happen at the membrane, right? This receptor will activate something else, and then that protein will activate a bunch of other proteins. This is called signal amplification, right? Or signal transduction. This means that a cell can detect a signal that is very low strength on the outside and actually respond to it, right? So, a cell might bind, I don't know, like I'm making up a number, but 100 molecules of growth factor. And still, even though 100 molecules is not that much in the context of an actual cell, we'll still be able to produce an output because it can amplify the signal. So some of you engineers out there, what might you refer to that idea as? I'm wondering if you're seeing connections to your own fields, because I know that there are engineers out there, right? This is a process that can take a low strength signal and amplify it, which is changing what? For some cross-disciplinary language. An amplifier? That's not the word that I was looking for exactly. Signal amplification, that's it. I was more thinking about maybe you can correct me if I'm wrong. Maybe I don't have the correct term for this, but I like to think of this as changing the gain. It's the same input, but the output is louder because you can amplify that signal. All of you are correct. It is simple signal amplification, but I was looking for like a parallel to a different field. That weird redstone thing from the block game. Yeah, I mean, true. You could propagate a redstone signal just like this, because redstone signals are circuits the same way that cells are a collection of circuits as well. True, gain is another keyword that's applicable. Okay, good. That's what I thought gain was as well. I was aiming for an electrical engineering parallel gain. Yeah, cells have gain too, right? And it is this signal amplification that allows it to do this. So if we go back to the real pathway here, a lot of these things in the middle are just amplifying other things that will make the signal louder and eventually propagate to the right area. Okay. So eventually that signal gets loud enough, and that will activate certain proteins that can then enter the actual nucleus itself. And so these are called transcription factors, right? So you have a series of chemical reactions that amplifies and that generates a signal that is loud enough to cause chemical reactions on these specific proteins that will enter the cell and activate a response, right? This protein will bind to a certain region of DNA, which will then make it easier for an RNA polymerase to bind. That RNA will be made, and then that RNA will be made into a protein. So this is kind of a complete circuit, right? Binding to a receptor causes a propagation of signals in the complicated circuit known as the cell, which will then shove a specific protein into the nucleus. That specific protein will bind to DNA, bring with it the thing that makes RNA. RNA gets made, and then protein gets made afterwards. This reminds me of using cell regulation pathways as logic gates. Yeah, again, like I'm okay with certain tech-y simplifications of the cell, and I think thinking of cells as logic gates is perfectly okay, perfectly good to think of them as logic gates. Like they are capable of doing some very intense computation, which makes sense, right? Like cells have to be insane computers for them to support life, because life is a series of very complicated inputs, and to be able to respond to that in an intelligent way, it's no wonder the cell is so beautiful and so complicated. You have no amplification with logic gates. I think it's like two separate ideas, right? Like the cell as a unit has to be able to compute something, and when cells act sorry, let me back up. Internally, the cell is amplifying signals, but at the layer of the receptor level, you can have logic gates. I don't know about cells as a whole, just because I've done less engineering, but certainly you can have logic gates by different combinations of receptors being near each other on the cell surface, which I think is really, really interesting. So I think you can do both. All right. So I want to dive deeper, not super deep, because because if you've recently taken cell biology, you can do mitosis way better than I could right now, because it's been too long since I even thought about a cyclin. But I want to have a little bit more depth into the cell cycle, because it's a big part of cell biology. It's also a very complicated dance that all of these molecules have to do in order to do this properly. It is so essential for life, etc., etc. And we could talk about like how mitosis happens, but I kind of don't want to do that, right? Like I don't want to talk about how the spindle forms. I don't want to talk about when DNA replicates. What I actually want to talk about, because I'm more interested in it, is the regulation of the cell cycle. So what is the cell cycle? in order to support life, right, you have to replace old cells, you have to grow certain organs, etc., etc. This is all governed by signals that tell a cell to divide, right? This is called a cell cycle. This includes mitosis. If you are a stem cell or a replication competent cell, it is meiosis temporarily for if you are a sperm or an egg. But instead of talking about how the cell cycle works, I would rather talk about how it is regulated. When is a cell allowed to do this? Because I think if you are digging down into interesting research problems like longevity and cancer, this is really, really important for you to understand. CDK, yeah, General Mustache. We're going to talk. I don't want to get too in the deep with CDKs. I'm just going to talk about cyclins, actually, cyclins. Watching Phi while writing an abstract for Congress. Truly biology maxing. That's amazing. I'm so curious as to what you're writing a Congress about. That's very, very exciting. I don't even remember the names of each cell cycle's phases, and I've read stuff about it lots of times. Yeah, that's like the level that I went into before I had to refresh for this stream, right? I'm glad to do these 101s because I can reteach myself some things, but I don't reteach myself all that much, right? I just need a little bit of a refresher. Okay, I want to talk about cyclins, because this is such a fun and cool story within the vignette of the cell cycle, right? Because again, I only like to talk about things that are cool and interesting to me. If you're a student, you're going to learn about how this actually works. This is for fun, okay? In 1982, a scientist named Sir Timothy Hunt was studying fertilized sea urchin eggs, okay? And he radiolabeled the eggs with methionine, radioactive methionine, and then extracted the proteins and separated them by size, right? And he noticed that one of the bands was disappearing. Well, it rose before mitosis and then disappeared afterwards. And he called that protein cyclin. And he named it after the fact that he really enjoyed cycling at the time. He had no idea what this protein actually did. This is a direct quote from him. By the way, the name cyclin, which I coined, was really a joke. It's because I liked cycling so much at the time. But they did come and go in the cell. So it's kind of, you know, it's fortunate here because this is a protein that governs the actual cell cycle. And Marlon is saying that cyclins are cool. They are cool. I don't remember about how they work all that well, but I do remember being very, very impressed. So how do they work? It turns out that even though he kind of just joked about naming it cyclin, it was a very, very appropriate name because this is a protein that controls... Oh, by the way, I forgot to mention this here. Yeah, he won the Nobel Prize for this, right? Nobel Prize in Physiology and Medicine. This is a major, major discovery, the discovery of these cyclins. So on the left here is cyclin D, which is a specific type of cyclin that is responsible for kicking off the G1/S transition. This is the, you could think of it as the first stop in the cell cycle, getting ready to replicate the DNA within the cell after the cell is done growing. So how are these proteins? How do these cyclins, how do you actually, molecularly, how do you tell a cell to begin this very complicated multi-step process, right? Because, you know, moving things in and out of the cell, I think it's pretty, you can kind of get a handle for how that works. I think for the cell cycle, it has to be something quite abstract because it happens in steps, right? Let's look back at this diagram here, right? Let's see, we have mitosis, right, where the cell actually splits, and then we have G1 where the cell is growing, S where it's preparing to replicate its DNA, G2 where it does more growing, and then splitting off again, right? Like I probably butchered that order. Actually, no, that's pretty good, right? G1, and then you have DNA synthesis, so you have growth, and then you have replicating your DNA, you have another stage of growth, and then you have the actual split, and then you're back at G1, right? A multi-step process that has to be carefully regulated, right? You cannot have this thing go wrong, and if it does go wrong, you have to abort right away. And I thought they named it cyclin because they're in circular form. That would have been another fun way to describe them, but again, they don't really look all that circular. Okay, so how do these cyclical proteins, and by cyclical I mean they rise and they fall at different stages of the cell cycle, how do they actually govern the cell cycle? And you might actually get a hint just by looking at these, right? I ruined it. I was gonna have chat answer this, but you can kind of tell. I mean, it's actually still not entirely clear. So here is the concentration of these different cyclins mapped to their timing in the cell cycle process. So if you know the answer here, then keep your answer to yourself. I want the non-biologist to kind of just look at this depiction of data and guess what is the signal to actually move from cell cycle step to cell cycle step. Right? What does this actually look like? If you had to guess, how do we go from G1 to S? What is the signal that actually pushes us from one cellular program to the next? Day-night cycle? It is certainly a cycle. Not necessarily pegged to day and night, though. Use the figure on screen, the cyclins and their correspondence to the stage of the cell cycle. So G1 is a phase where you grow the cell. S is the phase where you synthesize new DNA. G2 is another growth phase, and then you have mitosis, which splits the cell. So what governs, what looks like it pushes us into the next phase? That's this concentration of D, cyclin D. Yeah, that certainly looks interesting, right? It looks like that, you know, as cyclin D increases, that when it gets kind of high, we get shoved into the S phase. Isn't that interesting? Age? Not quite either, because even as you get older, your cells still have to divide, right? And age is correlated, right? How long a cell has been around is correlated to whether or not they will divide, but not always. Yeah. I guess the reaction kinetics of the involved proteins will scale up and down the amount of time each cycle takes. Yeah. This is governed by reaction kinetics for sure. What happens if a cyclin cycle processes too quickly so that the chromosomes aren't done replicated? Oh, we will talk about that in just a little bit. But yeah, I'll go with the math's answer. Concentration of D, when D is high, it looks like we get shoved into S phase. But also, when D is high, look at the correlation, right? As D increases, you also see an increase in cyclin E, and it looks like when cyclin E is at its zenith, that's when the proper S phase actually begins, and then we have a huge cyclin E drop off. Similarly for G2, it looks like, you know, we have high cyclin D here, but we also have a gradual increase of cyclin A, and it looks like when we have a lot of cyclin A, we get shoved into G2. And similarly for cyclin B, it looks like when cyclin B is very high, we get shoved into mitosis. Now, what's the problem with this explanation? What's the problem? I just said cyclin D is high, and so we enter S phase. Cyclin A is high, and so we enter G2. Cyclin B is high, so we enter mitosis. Why is that a problematic explanation here? Why is that problematic? It's kind of weird that cyclin D is present for each stage of the cycle. Can cells abort the cycle in the middle by getting rid of cyclin D? And also a very, very interesting question that I will address in just a bit. It seems like cyclin D is eliminated very slowly. Yeah. Looks like it, isn't it? Works well for cyclin E, but the others accumulate more slowly. Yeah, it doesn't quite match up. It's also correlation, right? I just said, you know, if you were a scientist back in the 80s and you just were studying cyclin levels, you might notice these patterns and think, oh, cyclin E could govern the transition in S phase, but until you actually test it, it's not entirely clear. So I will say for now that these cyclin levels are correlated with entry into the next phase, right? And it's not super tight. Cyclin E is super tight, but the rest are not super tight, which is pretty interesting, right? But we do see that each of these cyclins have a different behavior, which I think is very, very interesting. And here's where we'll talk about how this system actually works. So what's going on under the hood? How does this actually govern whether or not the cell transitions from one stage to the other? This is, of course, way too complicated to do in one stream. This is a week in your cell biology class. But here's the overall concept, right? You have these stages, and these cyclins indirectly regulate each other, right? So Marlon is talking about reaction rates and these interlocking signals. And again, this is an omega super simplification. Please do not say this on your exams. You will get things wrong. But basically, if a cell has to divide, it needs a growth signal. And so this growth signal binds to the cell membrane, propagates the signal, then you get the formation of cyclin D. Cyclin D activates cyclin E. And we see that here, right? As cyclin D increases, cyclin E, it lags behind because you need, cyclin D is ramping up. So over time, that signal will reach cyclin E. So D is upregulated in cyclin E peaks, and this peak activity does a whole bunch of biology stuff that it formally transitions into S phase. But then look at this, cyclin E actually pumps the brake on cyclin D, right? They regulate each other. So when cyclin E, as you see the increase in cyclin E, you actually see the growth rate of cyclin D decreasing, right? This isn't a linear thing. It's very fast at first, but as you accumulate cyclin E, you see that the growth of cyclin D is coming to an end, right? This curve is slowing down. As cyclin E is high, it activates cyclin A. So as you can see here, cyclin E is up, and then there's a little bit of a lag, but cyclin A starts to accumulate. And look at that, cyclin A also negatively regulates cyclin E. So once you get an accumulation of cyclin E, that's when cyclin E drops off, right? Isn't that cool? These cyclins all regulate each other. And look, as cyclin A increases, then you get a little bit of lag time, and then cyclin B starts to increase. Cyclin B negatively regulates cyclin A, so as cyclin B increases, you get a drop off in cyclin A. And we know that when you have high cyclin D, you get shoved into, you get this signal to start the cell cycle. When you have high cyclin E, you get shoved into S phase. When you have high cyclin A, you get shoved into G2. And then when you have high cyclin B, you get shoved into mitosis, right? But these checkpoints are governed internally, right? There's not one thing that is central that regulates all of them at the same time. They regulate each other. And this is how you can get cyclical activity. And this is internally conserved, right? And I'm using that term in a sense where if you no longer have growth signal, then you don't have cyclin D, right? Then if cyclin D isn't high, then none of this actually happens. This is really, really cool. I hope I did an okay job at explaining that. I'll actually put in a poll here to see if that makes sense. Did that make sense? I can explain it one more time, but try to, the arrow and the arrowhead is activating, and then the flat arrowhead is inhibiting, so it breaks it down. So cyclin E, for example, will break down cyclin D. Had to run another analysis and miss the entire explanation. No problem. I'll do it one more time just because I think it's really, really cool. So to regulate the cell cycle, you need something that is very, very carefully maintained, right? Something that is very sensitive and needs to be able to shove the cell through a multi-step process all at once. And if you don't do this successfully, you need to make sure that the cell then dies, right? So there has to be thresholds that are met in order for the cell to successfully make it into the next stage of the cell cycle. And so this is achieved by the accumulation of cyclins, right? The thing that governs the transition between G1 and S phase of the cell cycle is cyclin E. G2, S to G2 is cyclin A, and then mitosis is cyclin B. When these are high, that's when you get that trigger. When you get a growth signal, you get an accumulation of cyclin D. Cyclin D activates cyclin E. That's why you see this green curve lag behind the red curve. But as you get an increase in cyclin E, you see a breakdown of cyclin D because cyclin E negatively regulates cyclin D. That's why this is fast at first and then it gets slower over time. Cyclin E, when it's high, activates cyclin A, which is why you have a lag time between the blue line and the green line. Cyclin A starts to increase, and when you have high cyclin A, you get the S to G2 transition. But cyclin A, just like its predecessor, indirectly breaks down cyclin E, so when the blue line increases, you have a decrease in the green line. Similarly, when cyclin A is high, that activates cyclin B. That's why you have this lag. And then when cyclin B is really high, you get the shove into mitosis. And again, as cyclin B increases, you have a decrease in cyclin A. This is all beautifully maintained and consistent internally. And if you don't reach these checkpoints, that means something bad has happened. You actually need a certain threshold to be reached for the signals to get loud enough for you to actually activate these programs, right? So I think Marlon had a question about thresholds before. Build up to a threshold until a state change occurs. Exactly, yeah. You need a certain threshold for this to actually happen. What if a cyclin cycle processes too quickly so that the chromosomes aren't done replicating? That's why you have all these checkpoints, right? If there is, I don't know for certain the answer to that question, Marlon, because again, that's a little too much detail that I was prepared for today, but there are I think there are proteins that communicate between the nucleus and outside the nucleus that tells whether or not the replication is finished. I don't remember how they work though. Yeah. Maybe once the cell is in the cell cycle, growth factors are ignored, so no more cyclin D is made. Well, it kind of depends. If the cell needs to be constantly replicating, you might always have that growth signal, but in order to because the growth signal comes from the outside, right? That growth signal might be there, but if it's going through these processes, like cyclin E is knocking down cyclin D. So even though the signal is there for cyclin D to be upregulated, there's so much cyclin E that it prevents it from doing anything strange, right? That's why this is so nice. Even in the process of additional growth factor, it needs to finish before cyclin D is permitted to be high again. All right. So it's been a long time since I actively studied the cell cycle. This is pretty much the only thing that I remembered in any detail is this system of cyclins because I thought it was very, very beautiful, right? This system of things regulating each other. It's like a beautifully connected network, and they dance together. This is kind of a very elegant dance, and the steps have to all be done correctly. Otherwise, you should not be allowed to proceed to the next step, right? Because catastrophic things can happen if you divide without being ready. Should this tickle any programmers / engineers' fancies? I think this is just a series of if-then statements, right? If threshold is met, then activate this program. And I think that's a fun way to think about the cell. Okay, I'm going to take a break here and take a drink of my tea, because this is the last leg of the journal, the 101 session. I'll pause here for any general questions you might have about cell biology. I can't promise that I'll be able to answer any of them, or any comments that you have about what you've learned so far. If something that you thought was particularly cool about what I've talked about, I would love to hear that. Or if you have any additional questions before I get into some more modern cellular biology. Oscillators might be more comparable, especially, yeah, oscillators, that makes sense, right? These states are oscillating within the cell. And actually for developmental biology, oscillation is really, really important because sometimes it's not even as simple, although the threshold thing isn't even that simple to begin with, but sometimes it's not even as simple as, oh, there's enough of the signal. Sometimes oscillation is the signal. Sometimes it's not about, oh, how much of the molecule is around, but what is the frequency of the pulse of the signal? And sometimes that can communicate specific information with regards to how a cell develops, which I think is just incredible. I think that's going to be a really fun one for engineers out there, those of you who are engineer-minded. Must have been crazy being a biologist in the 70s to 80s. I think it must have been crazy to be a biologist anytime after the 50s, honestly. Like well, I mean, I have bias, right? I'm not an animal person. I'm a cell and molecular biologist. So I think that from the birth of molecular biology, arguably in the 30s to 40s, from then on, it's just been a nonstop, like, screaming developmental process of figuring out things about the cell. Although I think a lot of the foundational work in developmental biology, when I talk about it, is just shocking. It is so... Like, if you thought that the things in cell bio were cool because of how complicated these machines can get, think about machines coordinating other machines, and that's the developing embryo, right? I think Kyrgyzstan actually just released a video about how insane pregnancy is. I haven't watched it. I actually hate watching Kyrgyzstan videos because they're so good and makes me feel bad about myself. So I'm probably not going to watch it, but I'm sure it's great. I'm pretty sure it's great. All right. And I think a part of like a good education in biology, especially at the early stages, I know some of you have yet to take your first real class, is making sure that students are always aware of how much left there is to know, right? And I can't put that on a lot of American teachers' plates because they already deal with so much. But if you can find the tiny bit of room to look into modern research, it might help some of the more passionate students find a passion for biology, right? Because I think a disservice that was true for me learning AP biology in high school was getting the feeling that it was already done, right? I remember the distinct feeling that I'm like, oh my gosh, there's so much that we already know about the cell. We already know how, the cell cycle happens. We already know X, Y, and Z. Where is the room for the little guy like me? How could I ever contribute to something that is already so fundamentally well understood? And I think that's just because in high school, there's just so many other things for you to worry about, your teacher and you, that there's not really room to squeeze out modern research. And that is a service that I would like to provide with my streams, right? This is what I think is the actual useful part for students. Before, it's a little bit of an overview, a little bit of a teaser, just for fun, vaguely educational. But this is, I think, an actual important thing to remind people that there are still a lot of earth-shattering discoveries that we have yet to make, and there is still so much surface area for a young researcher to get into. It might seem kind of intimidating because you have to learn everything that everybody before you had done in order for you to do the new thing, and that can feel kind of discouraging. But like, the thing is as research continues, your reliance on some of the earlier stuff will fade and your reliance on some of the newer stuff will strengthen, right? Like, I didn't have to learn a lot about, I certainly didn't have to learn a ton of foundational molecular biology to get to where I am now. I mean, it was a lot, but it wasn't everything, right? Your teachers will get better over time, picking and choosing the things that are actually going to make you a better current researcher. And I think that understanding that there are still some things that are supposedly somewhat basic that are just waiting for you out there is pretty good, I think. Pretty good. Having watched the Kirkazad video, it was very informative. I can imagine they're so good, dude. I applied to work for them, but they only take German speakers, so I don't feel that way. They're a huge team of animators and everything. I think, I joked about them making me feel bad, but it's like, what can I do that Kirkazad can't, right? That is how I have to think about all of my videos. And like, it's not good to just do Kirkazad, but worse. You have to do something entirely different, and that is my own channel. And I know that I have my own identity that Kirkazad can't, because Kirkazad is a team of animators. They don't have one centralizing personality, but it is like, man, it takes... having more than two people work on a project, because Toxo like draws some assets for the thing and then I do everything by hand, is just like so frustrating. So when a Kirkazad video is very good, I'm like, oh my god, it would take me like 18 years to make something of the same quality. So I have to cover content a bit differently. Unrelated, but I gotta talk about how little I care for evolution. Like, everyone's talking about the beginning of life in the first cell and all I care about for protein machines. The next video I make talks a lot about evolution, and it is something, I think it'll change your mind about your view on evolution. Ooh, do I tease it here? I think this is a good time to tease it, just because I think you'll find this interesting. The next video is on Yersinia pestis, the Black Plague, the Black Death. It is a video that I have had long aspirations for covering, but I wanted to wait until I was good enough to cover it, right? Because if you looked at my rabies video, I like it, it's fine, but I've gotten a lot better at telling stories since then. And I've been very, very excited to tell the story of Yersinia pestis. And a little teaser for all of you, Yersinia's ancestors were gut pathogens and heavily environmental. Yersinia pestis exploded onto the scene 6,000 years ago, and its cousins and mother and father are nowhere near as deadly. And if you think that's interesting, well then go watch my video when it comes out in a couple weeks. That's interesting, isn't it? And that's an evolution question. How did Yersinia pestis evolve to be one of the deadliest bacteria to ever exist? Not by total kill count, but by case fatality rate, right? One of the deadliest bacteria to ever exist. It didn't just pop out of nowhere, it evolved. And so hopefully I've piqued your interest. Evolution is very interesting. It just depends on what you're interested in. Yeah. So that's a little teaser up ahead. You'll think you'll do a video on the newly discovered hemifusosomes? I'm not entirely sure. Probably maybe for a journal club, because I think new organelles and stuff are neat. Let's talk about kleptoplasts. One day, I'm saving kleptoplasts for its own stream. So if you don't know, in my Discord, I have a channel where I just hoard papers that I'll cover eventually for a journal club or maybe a video. And the kleptosome is an organelle that is designed to steal chloroplasts from plants, which is insane. Another very cool candidate that I could have had here, but I want to wait until I can dedicate more time to that thing. Waiting for people to invent the zap knowledge straight into your brain innator. I mean, part of the fun is the struggle, right? I don't think... Okay, so why is anything interesting to learn? There is some challenge. There's some process that I know you meant this in jest, of course, but I think it's an interesting opportunity to vamp a little bit about the joy of learning, right? Like, I get so much joy doing research and writing for my own videos because I'm reading primary sources and then chewing it in my own brain, and I'm tasting the material firsthand. And you know, I like the videos that I make, but you are getting, you know, I'm like mama birding it for you, right? I'm mama birding it. And the video that I make is my mama bird regurgitation, which is, I think, still good, right? You know, baby birds can't chew the food on their own, so they need a bit of that regurgitation. But when you really get into it and you struggle with it, there's a taste to primary scientific literature that is unreal. Like, you know, as much as you may enjoy my videos, I enjoy my videos way more than you do. How egotistical is that? But it's true, right? The taste that I get from when I'm learning about Yersinia pestis, it is delicious. It is delicious. And I wish I could beam the feeling of discovery into your brains, because it is only by struggling through these papers and interpreting them that it actually like feels good. And like, for you, the regurgitation mommy bird video is still much harder. It is still much closer to the primary literature than what most other YouTube channels do. So I'm not like talking down to you. I'm just acknowledging that the more struggle you have with the information, the better it tastes. That's all. Please continue to enjoy my mama bird regurgitation. I make it very delicately for all of you. Winning Nobel Prize for biology or making videos like Kyrgyzstan? What's more important? Well, I mean, I would like to make videos. That is my passion here. I was suspecting that for some time, given you said it was a popular disease. Yeah, it's been a long time coming. I felt like you were avoiding confirming it when I guessed it. I was totally avoiding it. I was totally avoiding it. Learning is fun until there's an exam. Exactly. Exams take a lot of the fun out of it. But when you get to do it for fun, like I do, thank you to members, and trying to make a YouTube career work, it is unparalleled joy. I think it's so much fun. You know, creators talk about burnout a lot. I think if I can do YouTube like this forever, that would just make me the happiest cell on the planet. Yeah. All right. So I'll have one more sip of my tea, and then let's talk about some brand new cell biology. This came out in February of this year. Sounds more fun than struggling to get cells to grow. Struggling to get neurons to grow is terrible. If you are working with immortalized cell lines, I envy you, because when I did my graduate research, I had to work in post-mitotic cells that did not do the cell cycle. And so if you screw up, you are just screwed forever. All right. So, new organelle-ish thing just dropped, the blebosome. So what the heck is the blebosome, right? What's this new cell biology about? These are absolute units of extracellular vesicles, right? Here you have a cancer cell, a mouse tumor cell line, and here we have a blebosome. This is a vesicle, and now that we have Cell Bio 101 under our belts, you know that this is something that is secreted from the cell, and it's enormous. It's huge, right? Compared to the entire cell, this is an enormous chunk. This is absolutely crazy. The largest blebosomes can be up to 50 microns in diameter. That's 3 neutrophils across, right? These blebosomes coming off of these cancer cells can dwarf immune cells in their entirety. I have never seen an extracellular vesicle this big before, right? For context, the large extracellular vesicles, the land V's that I mentioned in my neutrophil video, those are a micron in diameter, and I already called that pretty big, right? I already called those pretty big for a neutrophil. These are absolutely gargantuan, huge. So what are some properties about these blobosomes? Well, weirdly enough, they have mitochondria in them, right? Maybe that's not that weird. They're so big compared to the cell that maybe it's not unusual that they just have mitochondria packed in them by accident, right? If these things are coming off of cancer cells here. These blobosomes are so big that they're almost like miniature cells in of themselves. Not only do they have mitochondria, as outlined here in blue, they have their own endoplasmic reticulum. They have their own autophagosomes, which is part of the Golgi system. They have their own endosomes, another part of the Golgi system, their own lysosomes, acidic packets of enzymes that chew up proteins and recycle them. And they also have, well, they're made of actin, so this is not that unusual, but the blebosome themselves, they come with their own stuff. These are huge, right? It's an entire organelle larger than certain cells. Yeah, exactly, Seed. They're insane. Organelles with organelles, yes. These blebosomes can also eat up other extracellular vesicles, right? This is like the Katamari of extracellular vesicles. These here in green, these are labeled extracellular vesicles that the researchers added to blebosomes. Blebosomes labeled in purple. And what they found was that these blebosomes were able to eat these labeled extracellular vesicles. They can eat other vesicles. That's how big they are. That's insane. Absolutely insane. I thought I was looking at a cell and looking for a blebosome. Yeah. Is a cell inside of a cell? It's not proper to call them a true cell, though. They are exported from a cell, but they are as big as some cells, and they have their organelles. It's stupid, crazy. Which types of cells have them? Cancer cells. Cancer cells make these. Cancer cells. So blebosomes can also excrete their own extracellular vesicles, right? So not only can blebosomes eat other extracellular vesicles, they also can spit out their own extracellular vesicles. These are extracellular vesicles within extracellular vesicles, right? So, you know, I heard you liked them, so here they are, right? You can see that these extracellular vesicles in this lane of the Western blot, you can see that the stuff that comes out of the blebosome has the stuff that you would expect an extracellular vesicle to have. Absolutely crazy. These are nuts. The blebosomes seem almost cell-like, although I don't think they have a nucleus. They don't have a nucleus. Although you can say that red blood cells are a cell, but they also don't have a nucleus, right? What stuff are blebosomes missing that it needs to be a cell? Here's where I would say that, you know, say it with me, everybody, biology is a spectrum. We just don't consider blebosomes cells because they don't, I don't think we call them cells because they don't arise the way a typical cell would arise, either from differentiation of a stem cell or through mitosis. They branch off, they bleb off of an existing cell. And unlike yeast that do budding, they are not like replication competent, right? Although, yeah, I mean, I would argue they are pretty cell-like, but, you know, I would say that they are probably more appropriately classified as just a weird extracellular vesicle. Exclusively cancer cells, or are they in any other fast-producing cells? An excellent question, Nick. This paper is from February of 2025. We don't know, which means if some of you are reaching the end of your undergraduate career, you might be a part of the wave of people that discovers when this blebosome thing actually happens, right? We know that cancer cells do this. I don't think we know if any other cell does this, although you can read the paper linked in the description if you want the author's take on it, because I didn't read the paper in full. I only did the snippets for the stream. Please tell me it's a joke and not something serious, because it's close to surreal. Like the existence of the blebosome, I'm very sorry. It is very real and almost spooky. I don't know. It's weird. It's crazy. It's a little funky. It's nuts. Sorry for a lot of questions. Do I know what kind of cancer cells? Only the ones that they've tested, which I don't really recall. It's a mouse cancer cell line. And again, it's free to read if you go in the description. You can go to their method section and actually find out which cancer lines. And actually, this is also very fun. If you don't have biology experience, go to their methods and you can practice finding this information for yourself. They'll actually tell you which cancer lines they used. I just don't remember. Please tell me this is a joke because I don't want to isolate those chonkers. Why not? It seems fun. I've never done something like this, but the way that they do this is that they spin down the cellular contents of the centrifuge, and because they are so big, you can actually isolate the fraction that contains them, which is really, really fun and interesting. What existing pathways do blobosomes emerge from that cancer cells use? Don't know. Papers from 2025, baby. Brand new stuff. This is just a descriptive paper, just showing what are the properties of the blebosome. Of course, I would love to know how the cancer cell actually forms this, but again, that's why this is so exciting, right? This is so new that these very fundamental questions that chat, which is 50% biologists, 50% non-biologists, the fundamental questions are so new that even non-biologists can ask them and have it be a legitimate research project, right? It's very, very interesting. All right. How did they discover them in the first place? I'm guessing they mentioned that in the intro that I skipped. Haha. Okay. So it's cool that these cancer cells make these blebosomes, but do they actually happen in the context of biology, or is it something that they just do when you isolate them, right? Because admittedly, up until this point in the paper, it's a very artificial system. At the very least, they happen when cells are in a 3D collagen, that should be collagen environment, which is somewhat physiological. So this is a video of a real cancer cell that is embedded in a 3D collagen matrix, much like what you would see in a real living body. And the arrow is pointing to the cell, and there's going to be a bleb that forms on top of this cell, and you can actually see the blebosome forming in real time. So we'll actually see pop. Watch that again. And these cells are, actually, I don't think it's this particular figure, but just want to keep on watching this and repeat. How they come up with the name is perfect. The bleb, so a bleb has a very precise meaning within biology. Sorry, I forgot to mention this just because it's easy to forget that it sounds like a nonsense word to most people, but a bleb is literally a membrane protrusion. It is what happens when a cell is perturbed. And, you know, blebs can be mentioned in the context of like apoptosis, which gets a lot of blebs, you know, when a cell is starting to fall apart because of programmed cell death. And the blebosome is one of these like enormous things that blebs off from the cell. Also, welcome, more juice. Welcome. Purified from real healthy or real pathogenic bone marrow? Oh, excellent question. I don't remember. I blazed through this paper in time for stream. That's an excellent thing to double check. And if someone can grab it, I can put it as a response in the comments of the section. Yeah, so these are huge, brand new extracellular vesicles discovered in the current year, whose function is not really totally well understood, but we do know that some cancer cells make them. So the platelets gone wrong? Don't know that for a fact. I'm not entirely sure. Marlon asked a very good question, and the answer is almost certainly within the paper as to whether or not this is some cancerous bone marrow or regular bone marrow. Such a cool video. Love this. Okay, so what does the blebosome do? Well, we don't really know yet, right? This is an exploratory paper just describing some of the properties of the blebosome, but something that this paper does discover is that the blebosomes contain a lot of immune checkpoint proteins, a lot of proteins that tame the immune system, which is pretty interesting. And so is there anybody in chat that might speculate why a cancer cell might be interested in stuffing these extracellular vesicles full of proteins that can tame the immune system? Why might that be in the interest of a cancer cell to do so? No way, it's an actual formal term. I actually don't know. Like some terms like Sonic Hedgehog, you can be like, okay, somebody more serious could have named Sonic Hedgehog better. I think blebosome is perfect, actually. There's not a less silly way to mention it, because bleb, when you hear the word bleb, it feels right? It's like Kiki and whatever, Kiki and Bobo or whatever, right? There's a bleh. There's a sound, there's a feeling to it, and it's very descriptive, right? Bleb, because that's what's happening to the membranes. Yeah. To not get killed by them? Yeah. To not get targeted by immune cells in the first place. Yeah, that's the hypothesis here, right? That these cancer cells might be cramming these enormous blebosomes full of immune checkpoint proteins that will potentially, eventually, tame the immune system somewhere. Now, I don't know if that is true. Maybe this is an accident, right? Maybe this is the cancer cell malfunctioning, and it's just part of the side effects of malfunctioning is just sloughing off enormous sections of yourself, maybe. Or because cancer is under one of the most oppressive evolutionary regimes in all of biology, a series of unfortunate events creates this very potent immune evasion tool. I don't know the answer. I'm very excited to find out though. And again, maybe one of you will find it out. That's very it's so new and so exciting. Very, very cool. Yeah. To be a bait for the wider immune system. Maybe. Like they wouldn't initiate apoptosis. Maybe. Who knows? It's just there's a lot of stuff in there and it's capable of taming the immune system. So I think all of these are excellent guesses. Do they have any sort of protease activity? Don't know yet. What are these proteins in the diagram responsible for? Excellent question. I don't remember. They're just described as immune checkpoint proteins, and I finished my slides like 15 minutes before the stream started, which is why I also started a bit late. So not entirely sure. But yeah, lovely paper to read. It's also a Nature paper. So if you have any biology background at all post-college or during college, it's somewhat readable, I think, if you want to read the abstract and read through the pictures. That would be, I think, accessible. It's not too crazy of a paper in terms of the language. The bone marrow section was from healthy 8-week-old female mice. That's also interesting. So that indicates that maybe stem cells are doing this too. It's maybe not surprising because cancer cells are stem-like. So maybe this is a feature of stem cells more generally. I think that's a pretty interesting hypothesis, although who knows? Okay. I think that's it for this paper. Very, very cool. We are nearing the end of stream. I still have 200 push-ups to do for the Side Array Bean Fund stream, so I will try to do more of those. Maybe I'll try to knock out another 100 while I just make chit chat. So before you leave though, please help me figure out what merch to make. In the pinned comment and in the channel description, there is a link that you can go to. And this is just a little poll to see what people are interested in for merch. I can't do a plushie, unfortunately. I would love to do a plushie, but I have to buy like 500 of them. I just don't have the funds for that right now, right? But these other ones have lower barriers of entry, and I would love to sell some merch, get some Phi stickers and magnets out there. All right. And with that, there's one last plug that I would like to do before moving into the chatting and push-up section of the stream. And that is fellow VCR member Z has been on hiatus for quite a bit of time, and she returns this Sunday with Baldi's Basics. So I think that's going to be quite a fun stream. Always love popping into a Z stream. And that's it. I'll take any questions from chat while I start 100 push-ups. You can talk about cell biology, you can talk about whatever, or leave questions for me or any general questions about my channel or biology in general. I would love to just chat in between the painful experience of having to do all of these push-ups that I did for Saturday's Bean Fund. I will be right back while I do those push-ups. Ooh, desk mats. That would be cool. I think that would be cool. I don't know whether or not the store that I'm working with would allow for that, though. Although I'd have to look into it. Can you do 100 squats next? Today is squat day. When Toxo gets back, we're maybe going to the gym. And today would be the day that I would do squats. But push-ups is what I promise, so push-ups is what I will do. So be right back while I do as many as I can, and then I'll read whatever questions or comments you have from chat. Or even, if you want to just talk about biology freely, that's cool too. So BRB. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. Oh. All right, so that's 25 knocked out immediately. Oh. I was sore for like a whole week after that day. I usually don't do that many. This has been delightfully informative as always. Thank you. Thank you so much for coming, Tenbu. The sounds of ATP being consumed. Exactly. My muscle fibers are going crazy. Yeah. Oh, I'm hungry too. Doing the 100 push-ups on an empty stomach is pretty rough. I feel like I can never get enough calories in a salad because that's what I had for lunch today. I feel like there's a ratio between like the consumption of food and satiation. I think is also a really interesting topic that I don't really have a lot of background in, but I feel like feeling full in the moment does not correlate well to how long I will feel full later. I guess that's not really groundbreaking, but a lot of things that people say are like stomach fillers, like high fiber stuff keeps you fuller for longer. I feel like salads, which are crammed full of fiber, I feel like they just disappear. I don't really feel like they stay around for all that long. Something to do with ghrelin. Yeah, ghrelin. The hunger. There's like a stretch receptor in the stomach, and then I think ghrelin. I don't think ghrelin is ghrelin activated by stretch receptors. I don't quite remember. I feel like at some point you might need to change your character to a Larry the Lobster equivalent of Neutrophil. That would be quite something. I was skimming through the paper, and blood basal isolation does not sound too hard. It really doesn't sound all that hard. Although, of course, like, you never know until you do it for yourself, right? But because they are so big, you can just spin them down. And if you know which fraction to isolate and know what markers to test for, then it should be okay. Because I think what they did was they just spun it down and then isolated the stuff that had labeled organelles that weren't cells. So absence of nucleus and presence of mitochondria is your discrimination tool. And that honestly doesn't sound all that bad. Potato works well for filling. Yeah. Potatoes work pretty well, I would say. For me, the go-to is rice. I love rice, but rice is not good for you. And so I've been trying to eat better. Did you draw your own avatar? My wife drew these. All of the aesthetics of Five Neutrophil were made by Toxo, and I love them very much. My wife, yeah, I can finally say that. It doesn't feel like a meal without rice for me most of the time. It took a long time to, like, I've been moving. So I had a whole thing where, like, I spent several years on brown rice because of the purported benefits of, you know, having additional fiber and a better glycemic index, et cetera, et cetera. But then I worked with professors who were doing research on arsenic and DNA damage, and brown rice, depending on the area it's grown, can accumulate quite a bit of arsenic. Arsenic's not great because it interferes with DNA repair, and so I was like, oh, but I just got used to brown rice. So then I transitioned off of brown rice back onto white rice. But again, white rice isn't good for you either, so I've been experimenting with a lot of other alternate sources of calories, put it that way. Brown rice just tastes better. It was very much acquired, and I actually found that I missed brown rice when I switched back to white, just because I think it's kind of hard to replicate the steamed rice taste from like an actual good like Chinese restaurant, because I don't know, doing it at home, even though I have a nice Zojirushi rice cooker, it's not quite the same. Like, I don't know what brand of jasmine rice they're cooking up in those restaurants, but it's not the one that I buy, and I am continually on the search to mimic that exact same flavor. But isn't white rice made of brown rice though? So the arsenic is accumulated in the hull of the rice, and if you have brown rice, the hull is kept intact, which is also where all those nutrients and fibers live. But along with those nutrients and fibers, you have arsenic, which is no good. Is there like a level of arsenic exposure that your body can safely eliminate or ignore? It's basically always bad. I don't think the WHO has a safe lower limit for arsenic, which is kind of annoying. Yeah, I mean, it has hypothesized to cram up DNA repair, which is pretty much a no-go. Devastating. What software do I use for my VTuber avatar? I've been thinking about getting into the Eddie VTube space. So it is VTube Studio. This is pretty much the Premiere is not really the right word, but the most common starting point is to use VTube Studio as long as you have a 2D rigged avatar. Right? If you want to do 3D stuff, then I don't remember what that one is called. But yeah, 2D is nice because I like the 2D aesthetic, but if I didn't have Toxel, I might have gone with 3D because it's cheaper. Oh, rice is the goat. Rice is the goat. I love rice. I love rice, man. When I was abstaining from rice, challenging myself to experiment with alternate grains. Oh man, sometimes all you want is rice. I've had a lot of like dietary changes, moving towards like a healthier, more sustainable plant-based diet, but like rice just hits so hard. Oh my god, rice is so good. Isn't there a rice or similar dish that if you cook it too bad, it makes a molecule that be carcinogenic? That reminds me of potatoes, but I don't really know what you're talking about. I've started mixing lentils in with white rice as a middle ground. They even cook the same in the zoji. Oh, that's actually a really good idea. Oh, that's good. I like that. I'll have to try that actually. I think it's the fluffiness of rice that makes it more flavorful. Maybe. I had a mini lab accident in the lab involving sodium arsenate, and the doctor told me I wasn't in danger. I mean, there's a difference between like exposure limit for like dietary stuff, where it's like, is there a safe level for you in the blood? And like, is this a medical emergency, right? If you have a cut and there's not an appreciable amount of it, then like it might not be worth treating you with chelators. You know, there's a bit of a trade-off there. I'm glad that you are deemed to be unharmed, and I don't want to like raise any alarm bells. Like, I think if the doctor said we don't have to treat you with chelators, you're probably good. That makes sense to me. Try adding a little oil or chicken stock into the rice cooker. I'm trying really hard to be plant-based. I think it's an interesting journey that I've had over the last couple of years where I think 5 out of the 7 days we don't eat any animal or meat products, occasionally eggs. Not for like, I mean, pseudo for ethical reasons, right? I don't know, I don't feel bad. I don't feel great. You know, it's funny working with mice. I find it much harder to eat animal every day, right? That's just because I had to take care of so many. Like I had to deal with a lot of death of the mice. And I think for some people they can get desensitized to it, but I was sensitized to animal death, and I just don't feel great about having so much of my diet reliant on animal protein. So that's no good. And I think if I weren't the type to be experimental with my diet, I would probably still be eating a lot more meat, but I actually kind of like figuring out what I can get away with in terms of plant-based substitutes. It's like an ongoing challenge, which I think people don't talk about enough, because some people say, you know, when they're selling veganism or vegetarianism, it's like, oh, this thing is just as good as meat, etc., etc. It's like, as someone who still eats meat, just not as frequently, yeah. I think you have to rewire your brain to not crave animal protein quite as much. I think there is something gradual about that process. Like you can't sell vegetarianism to a meat eater by claiming it's just as good. You might be able to say, if you just try over a couple of months to years, that you will come to love a plant-based diet. I think is much more reasonable and accurate for the majority of people. VTube Studio is free too. You only pay to remove their OC flying around at the bottom. That is true, but if you also pay for the additional DLC, you can do collabs for easy setup. Because collabing, it's difficult. And now they have DLC that makes it very, very easy, which I used in my last stream with Mylan, which is great. Oh, so much food talk. I'm so hungry. I'm so hungry. I actually like pasta. I love pasta too. I love pasta. I've been cooking a lot more pasta. I actually made pasta for the first time by hand the other day with wheat flour instead of semolina, and it turned out a lot like like Chinese noodles, which I was very surprised by. Although it shouldn't be surprising because a lot of Chinese noodles are wheat based, but I was aiming for the actual restaurant homemade feeling with like semolina, so I bought some semolina too. Yeah. Damn, now I'm hungry. It's 5 a.m. 5 a.m. It's so early. You should be in bed. What the heck? Speaking of plant-based tofu, I am a tofu enjoyer. Although, it's not new information. Something that every vegetarian, vegan says. You can't call tofu a meat substitute, but I would say that it's a good protein source, right? That's the more accurate version because tofu doesn't taste like meat at all. In fact, like you should be using tofu where it is culturally relevant to use tofu because those dishes have survived millennia, right? Mapo tofu, homestyle tofu, what else? And that's not to say that you can't experiment with tofu, but I would start with mapo tofu, homestyle tofu, salt and pepper tofu, all the good Asian dishes. Tofu equals to soy cheese. I've heard that take before. I think it's kind of crazy, but more power to you. Wait, does brown rice accumulate arsenic more, or is it more due to where it's grown? It stores it in the hull, so all brown rice, if it is exposed to arsenic, will accumulate it in the hull. There are regions of the world that are known to have higher arsenic in the groundwater and ergo higher arsenic in the hull, so it's tricky. Oh man, so hungry. Could not leave meat out of my diet. TBH takes a lot of discipline. It takes a lot of discipline to eliminate it entirely, right? To not even allow yourself a dietary option, like as a strict rule, I think is not super achievable for the majority of people on a practical level, right? Like food is such a comforting thing that you're gonna find it very difficult to argue people out of taking part in something that is comforting, especially when most of life is just putting a roof over your head and seeking comfort, right? It's a lot of what most of us can afford to do if you are in the majority of humanity. I think what is again more reasonable for most people is convincing them that they could eat more plants, and I think that's true, and I've been trying that, and the progress has been interesting, right? I made a concerted effort to cut a lot of meat out of my diet. I think less than 2 years ago, and I think I'm a much better. Oh, I have a rant about that. I think I'm a much better cook. Like fundamentally, I think I can make food much better now that I have forced myself to stop relying on meat. Just because I think this is maybe a hot take, but I think meat is such a shortcut to making a flavorful meal. It's such a shortcut. You can put most pieces of meat down onto a hot surface and salt it, and that thing will be good. Whereas for like vegan/vegetarian cuisine, you have to do so much to make it even approach how good just a salted piece of meat is. But I think the actual things that I can do in the kitchen, I have gotten much better at since having to teach myself how to do all sorts of different things. And also like, it's like learning curve too, right? The more you practice certain skills, the less effort they take. So not only am I more practiced in certain techniques, I also do them more so I can do them faster, right? There are more benefits than just health in terms of cutting out meat. Is you also force yourself to make vegetables delicious, which is not easy. I'm gonna get canceled by both sides, both plant eaters and meat eaters, but I'll fence in on this. I'm bullish on that perspective. I'm also trying vegetarian for ethical reasons. Transition to vegetarian is not easy. It's not. Like, I very much sympathize. Like, some people can cut meat out purely on an ethical, like, moral thing. But again, so much of humanity, so much of your life is just accumulating resources and then seeking comfort that it's really hard to give up comfort, right? You have to reprogram yourself to find comfort in other things. Vegan cooking is decidedly different, but the different ingredients have their own merits, although some more than others. Yeah, like, I think, there's this argument that has been made to me by meat eaters that is like, well, wouldn't every vegetarian dish be improved with the addition of meat? I would say not always. I would say that mapo tofu is an excellent example of something that you could get away with not adding meat. Actually, that's a bad example because usually there's ground pork in it or ground beef, and that usually is pretty good. What's an example that I can think of? Chinese cooking is a bad example because usually there's always some chicken flavoring or fish flavoring or oyster, shellfish, something or another. but I would say like okay, another like half-assed way to propose this is that like there are certain dishes where you don't want something too meat-like, right? MSG is good enough for a meat-like flavor in home cooking, right? If you're cooking for the queen, maybe you zhuzh it up, but for your everyday, like you can get away with a lot with MSG and garlic, you know, providing that same similar, and be careful, similar hit to the umami meatiness of meat. But again, being 1 foot in every domain, it's not a perfect substitute. It doesn't cut out the meat craving entirely, but it does make it so that on in most days of the week, I can make it through without you know, wanting a burger or something. Yeah, plenty of good reasons to go plant-based. Obviously, you don't have to hear it from me. I'm probably not the only person to make this argument, right? Have I tried fasting? I don't have the discipline nor the desire to optimize my life that much to try fasting, right? Fasting, I think, is something that could work for people, but it just doesn't align with my lifestyle. Let me some quinoa. Yeah, I've been trying, what is it? Oh gosh, bulgur wheat, farro, you know, all those things as alternate grains. Interesting sources of calories. Purportedly better for you than rice, but you know, if I'm looking for a comfort meal, it's always going to be something with rice, I think. Just slug away. Thank you now for the stream. Your vids are calming to listen to while drinking. Thank you so much for joining me. Tofu is a nice ingredient in its own way. It carries mapo tofu very, very well, and it fries beautifully, and you don't have to do all the pressing. You don't have to do all of that. You just have to be patient. Yeah, I think a lot of the trap that people get into with tofu is the pressing meme, right? Like, press the tofu down with a heavy surface for an hour, condense it, then it'll fry and crisp a lot better. Sure. But if you want to do sustainable home cooking stuff with tofu, you don't want to prepare an hour. You know, you get home at 5, what you either remember to press the tofu the night before, or you wait an hour to prepare your dinner, right? It's just not practical. You have a completely flat surface like a heavy cast iron. That's like those optimizations I don't think are very practical. What is practical is you buy a block of extra firm tofu, you cut it up, you pat it dry with a paper towel, and you fry it. And you just fry it until it gets crispy, right? You don't have to do anything fancy like cornstarch or flour. I mean, you can, but you don't have to, right? My style of cooking is very, because I cook every day, more or less, 5 to 6 days, like 5 days out of the week at least, I'm cooking, right? And I cook a different dish every day because I grew up on like grandparents that would cook every day, so I'm spoiled in that way. But if you're spending so much of your time dedicated to cooking a different dish every day, you don't want to deal with stuff like breading tofu, waiting for it to dry out and whatever, right? Just fry it, just be patient. And I think that will make it easier for meat eaters to not feel like vegetarian cooking is so difficult. Because when people say bread the thing and oil the thing, I'm like, man, that's not making the argument better. Because at the end of the day, you end up with a product that isn't as rich as a cut of meat with fat on it, and you made them do a lot of work. So I think that is not the best entry point, I think. Have I tried cheese-based alternative like paneer? I thought paneer is still cheese, right? Right. I have tried cashew cheese, and it was delicious. Genuinely on par with regular cheese, if you don't like the dairiness of cheese, which is kind of insane, right? Because cheese is dairy. But if you like the dankness of dairy, but you don't like the fermenty, salty, briny flavor of cheese, but you don't like the cheesy taste of cheese, cashew cheese is great because it had that like same fermenty, salty, briny flavor, but it didn't taste like cheese. Texture of cheese though. Ooh, the cashew butter was so good. Cashew cheese, I mean. Wheat flour, that's interesting. Isn't that like soba noodles? Maybe. I'm not quite as familiar with Japanese noodle ingredients. Ethiopian injera uses teff flour and it's delicious. I love injera. I tried making it myself and it was horrible because we moved away from a restaurant that was Ethiopian and I miss it every day. And we don't have one nearby, and me making my own injera was a nightmare. That's very difficult to do. Not something that, you know, I would drive all the way back there to to have that again instead of making it for myself, which is the next topic for the members only video, is the calculus required to determine whether or not you should be making your own stuff at home. Oh my god, so much food discussion. I'm so hungry. Oh, so hungry. Yeah, veggie simmered and dashi, they're quite delicious on its own. Silken tofu as well. That sounds good. Sounds good. Chemistry is a true shortcut. True. If you understand the principles that underlie food chemistry, you can do a lot in terms of vegan or vegetarian cooking. It's true, but you have to be like married to the. You have to treat it like kung fu, right? Like I say this a lot. Kung fu is not violent. It's not martial arts. It is self. It is what is it? Not self-determination, self-cultivation, right? You have to treat it like a hobby on its own, I think. What's your favorite non-meat protein source? I, for the first time, made my own seitan by hand, seitan. It is the high protein gluten that is extracted from washing wheat flour. Oh, it was amazing. I would say that tofu, beans, these are protein sources that are not substitutes for meat because they don't chew like meat, they don't tear like meat, they don't contain fat like meat would contain. But a piece of it has to be homemade. Do not buy this from the store. If you buy from the store, it's going to taste weird, have a weird aftertaste. I did not enjoy buying seitan from the store. Making it yourself is actually insane. It is so high effort. You have to spend so many hours dealing with this stupid product. But if you have the patience and you want to try what I consider to be the best, healthiest meat alternative, try making your own seitan. There's a chef out there who teaches you how to do it in a way where it actually mimics the muscle fiber structure of meat. Because again, tofu is just a block of protein, right? It doesn't chew or bite like meat. But seitan, if you stretch it, you can actually stretch the protein fibers to look like muscle fibers. And that stretching of the fibers is really important for it to chew like a muscle tissue would chew, right? And it is the only protein source that I think scratches the meat itch, which is like, 2 years into this experiment, it's quite late for me to have discovered. But again, in my defense, the grocery store versions are quite unpalatable, in my opinion. I fried it in a very like northern Chinese style of cooking, where I fried the seitan and I tossed it in a mixture of garlic powder, salt, MSG, sugar, cumin, Sichuan peppercorns, and then let it get crispy. Because it gets crispy, right? It's made of gluten. It can get wonderfully, deeply crusty. Right? I mean, you isolated it from bread. It's maybe not that surprising, but like it has the muscle fiber structure of meat. It crusts way better than I think a lot of traditional animal proteins do, because it is like so heavily just bread, right? It's made to be browned. So there are comparable advantages. And the other advantage of seitan is you can eat a lot of it without feeling bad, right? Because as much as I love a pork chop, I'm going to feel bad after I have one, right? If I have a steak, I'm going to feel bad if I have one. Seitan, dude, you can eat so much of it, right? It is like, I think that the joke from The Good Place is like eating frozen yogurt instead of ice cream. Preferring frozen yogurt because you can eat more of it. I think the same goes to seitan. You can eat a lot of it. You can go back for more bites of that experience, and A, have it be much better for you health wise, and B, have it be maybe not as maximally good as meat, right? Like there's nothing better to me than biting into a fat cap in a steak. Like you don't get the same high from the seitan, but the integral, right, area under the curve of a seitan dish, I think, is quite high, quite comparable to meat. So if there's one recommendation I have if you are trying to kick the habit of eating meat, is to make your own seitan and stretch it so that you can develop the meat fibers. Oh, I think Indian cuisine is a good example. Also a very good example of cuisine you should look to for inspiration. I think I have quite a bit of Indian ingredients in my cupboard as well. Chicken broth flavored stock was made by just combining amino acid powders. Yeah, I mean, nutritional yeast and MSG, that's pretty brothy on its own. I used to joke that like the broth that the E. coli grow in, LB Luria broth, smells a lot like ramen. I mean, people who have smelled it might disagree, but if you open your mind, the umami-ness is a lot like ramen broth. It is quite good. Quite good. People press tofu. I've never done that. It's such a waste of time. I've tried it and it doesn't really. It's such a scam. It's such a scam. I'm an undergrad in college studying life science and struggling a lot, especially with the lab practicals. This was not my first choice. I wanted to study zoology. Well, if something's not your first choice, it's always going to be harder just because you are forcing yourself to do something and it is not coming naturally to you. And as a formal lab instructor, I just have to say, I'm sorry. I'd be curious as to what you are struggling with. I would be able to help more if I knew exactly what you were struggling with there. Yeah. I'm going to go ahead and do another set of push-ups. I'll be right back. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. Oh, okay. All right, I'm back. Another 25 in the bank. Have you seen the research on the drug that gave back movement to spinal cord injuries? I have not. That is very interesting. Be curious to read that. Maybe add it to the infinite backlog. I'm reminded how certain vegetable protein things are just extruder pressed polymer, but I'm using amino acids instead of synthetic material. Yeah, I mean, I think the thing that made me finally want to make my own seitan is watching that video where the chef explained how they were mimicking the structure of meat, right? Because I've been interested in synthetic meat for a while because, again, like I don't want an animal to die so that I can have protein on my plate. And something that they stress a lot in lab-grown meat spaces or artificial meat is getting the fibers correct. And so because you can manipulate seitan to generate those fibers, it does end up a lot more like meat than you would expect. How many push-ups do I have left? 150. Are you like hot and ripped? Is that why you don't show your face? I am showing my face. What are you talking about? This you might not like it, but this is the ideal cellular body. Yeah. The thing I struggle with is preparing for the practicals. We are told what we will do, but not how, and we need to do that ourselves. Like, also writing the principles of an experiment is difficult for me. I don't recall ever taking a lab practical, if I'm being honest, because that's not how any of the lab classes I have taught operated. So I'm actually curious as to whether or not we are talking about the same class. Like, what is the actual lab class you're talking about? Because, yeah, the classic cell and molecular biology labs that I've taught don't have lab practicals. Especially because, like, you could do everything right, and sometimes biology will just not cooperate. Also, biology labs generally take a couple days, so I'm a little surprised as to what you're going to be tested on. Lab-grown meat or cultured meat, I would like to work for companies developing it. That would be so sick, like, honestly. I would be like first in line for an accessible lab-grown meat. Like, in terms of my price breakpoint, I'm okay with lab-grown meat being twice as expensive as regular meat. I would consider buying a product like that, right? Because and you're like meat costs in America are so heavily subsidized, it should cost way more, right? Because if you think about how many resources go into raising an animal, of course, meat is going to be artificially cheap for the consumer. But lab-grown meat, like, in order for me to want to buy it and have it be a part of like the regular purchasing decisions I make, I'd go up to twice as expensive, maybe even 3 times, depending, right? Like, I'm willing to drop money on this idea, and I think it would be, yeah, just incredible. If you like engineering, if you like thinking of structure and growth, you know, a lot of cell biology, I mean, you know, wrap it back to the topic at hand, right? Cell biology is cell biology engineering is what you would be doing in a lab-grown meat company. I have a lab practical for molecular biology this semester. What is with these lab practicals? What? That's so interesting. I have never been a part of a lab class that had a practical. Yeah, like what was even on in your practical? What could you do that is not a waste of class time? Because I think that lab practicals are a bit of a waste of time. I don't think that they are effective evaluation tools. Currently, it's biochemistry. We have to isolate protein from milk. I preferred the regular cellular biology. Even genetics was easier for me. Oh, interesting. I just don't see why you need to be tested in that kind of way, because in real life, you'll have a protocol, right? You just have to know how to do it. You have to know what to search how to do it. And that's, not, I don't know. I'm sorry. I don't have any advice because I never had to take or administer a lab practical. Just good luck. Sorry, that's not very helpful. If it's not as good, I'll skip lab meat. I'm willing to be an early adopter, right? Like, not all technology is perfect when it rolls out, right? Early protocols of like the iPad were enormous and people made fun of them, but there are true believers who believed in that product, and I'm a true believer in lab-grown meat. As long as it's reasonably affordable, I'm very willing to be amongst the first to test it. All right, I'm gonna do my next set of push-ups. I'll probably end stream soon after I finish this 100. I would like to finish, or maybe I just do the last 25. I think I'll do one more set of push-ups and then call that a stream, and I'll have 125 more to go. Oh, okay. Be right back. I'll feed Patrikios a bit. 16, 18, 21, 22. Oh, okay. All right. I am back. Thank you all for coming to stream. I don't think I'll finish the last 25 push-ups. Toxo just got home, so I'm gonna go hang out with her now. Thank you all for coming. Again, fill out that merch form if you want to help me decide what merch to design. And also Z is streaming on Sunday, which is very, very exciting. All right, so thank you all for coming. I think my next stream is going to be about drug design, which I believe I talked about on my channel yet, and I'm very excited for that. Until then, bye y'all. Bye. Bye.